Hello and welcome to The Sleepy Scientist. Tonight, before the body drifts too far into its own quiet maintenance mode, take a moment to let everyone know where you are listening from. Somewhere warm and sleepy, somewhere rainy and thoughtful, somewhere far too late at night to be making sensible life choices, or perhaps somewhere perfectly peaceful with a cup of tea slowly cooling nearby. And if you enjoy calm, thoughtful science videos that make the world feel a little stranger, a little clearer, and hopefully a little easier to fall asleep to, then please consider subscribing to the channel. A like on the video is also always appreciated, because it helps more sleepy minds find their way here, which is a surprisingly nice thing for a small button to do. And to everyone already subscribed, thank you. Your support helps keep these quiet journeys through science going, one gently curious topic at a time. Tonight's subject is something that is happening inside you right now. Not in a dramatic, alarming way. Nothing here requires sitting upright suddenly and checking whether all your limbs are still attached. They almost certainly are. But beneath the surface, in the soft hidden darkness of the body, an astonishing amount of repair work is always underway. Cells are being replaced. Tiny errors are being corrected. Immune cells are patrolling tissues with the calm seriousness of very small security guards. Blood vessels are adjusting. Proteins are being folded, unfolded, recycled, and rebuilt. Old cells are being cleared away. New cells are taking their place. And if there is a bruise, a scratch, a strained muscle, a healing bone, or even just the normal wear of another ordinary day, the body is already responding. The strange thing is how little of this is felt. A person can sit quietly in a chair, lie under a blanket, or drift towards sleep, while trillions of living cells carry out a constant act of maintenance. The body is not a machine that waits patiently until something breaks. It is more like a city at night, with repair crews moving through dim streets, lights glowing in small windows, pipes being checked, walls being patched, messages being sent, and a few biological workers probably wondering who left all this collagen lying around. From the outside, the body can seem still. From the inside, it is never still. A small cut begins to close before there has been much time to think about it. Blood thickens into a seal. Inflammation arrives, not as mistake, but as a controlled and necessary signal. Immune cells clear away damage. Skin cells crawl slowly across the wound. Fibers of collagen are laid down like scaffolding. Over time, the surface becomes whole again, perhaps with a scar left behind as a quiet record of the repair. A broken bone does something even more impressive. It does not simply glue itself together like a cracked plate. It grows living tissue, builds a temporary bridge, hardens it, and then slowly reshapes that bridge according to the forces placed upon it. Bone is not dead stone. It is alive, sensitive, and busy, constantly remodeling itself in response to the life being lived around it. Muscle repairs after effort. The immune system repairs after infection. The gut lining renews itself again and again. DNA is checked and corrected inside cells. Damaged proteins are dismantled and reused. Even sleep, which feels like the great pause in human existence, is more like a night shift for the body's maintenance systems. And yet the body is not perfect. It scars. It slows with age. It sometimes overreacts, underreacts, or repairs in ways that solve one problem while leaving another behind. Healing is not magic. It is biology. And biology is always made of trade-offs. Fast repair may leave a scar. Strong inflammation may cause swelling. A powerful immune response may need careful calming. The body survives not because it does everything perfectly, but because it does enough things well enough, often enough, for life to continue. So tonight, the question is not simply how the body heals a wound. The deeper question is how a living body keeps itself together at all. How does skin know how to close? How does blood know when to clot and when not to? How does bone rebuild itself? Why do some animals regrow limbs while humans mostly form scars? Why does sleep help recovery? And what is happening inside the cells themselves, in places far too small to see, where the quiet work of repair never really stops? This is the science of how the human body repairs itself, and it begins with a simple, slightly comforting truth. Even when you are resting, even when you are doing absolutely nothing useful, the body is still hard at work, gently trying to keep you alive. The human body can seem, from the outside, like something finished. A hand resting on a blanket, a face softened by sleep, a chest rising and falling with slow, ordinary breath. A person sitting quietly in a chair, doing nothing particularly dramatic, heroic, or likely to be turned into a documentary. From a distance, the body can look still, complete, almost settled. But the body is not a finished object. It is a living maintenance system. Even in the quietest moments, it is repairing, replacing, cleaning, defending, balancing, and adjusting itself. Cells are being renewed. Old proteins are being broken apart and reused. Tiny injuries are being managed before they are ever noticed. Blood chemistry is being kept within narrow limits. Immune cells are moving through tissues checking for trouble. Skin is shedding and rebuilding. Bone is remodeling. The body is less like a statue and more like a city at night where most of the lights are low, but the repair crews are still out there somewhere, fixing pipes and pretending they definitely know where the leak is. This constant work is not a side activity. It is the condition that allows life to continue. One of the most important principles behind it is homeostasis. Homeostasis is the body's ability to maintain stable internal conditions, even while the world around it and inside it keeps changing. It does not mean the body is frozen in perfect balance. It means the body is constantly making small corrections to stay within a range where cells can survive and function. Temperature has to be regulated, blood sugar has to be managed, water and salt levels must stay balanced. Oxygen needs to reach tissues, carbon dioxide has to be carried away. Blood pressure cannot drift without consequence. The acidity of the blood has to remain tightly controlled. Every living cell depends on this hidden stability, even though the person walking around with all these cells may mostly be thinking about whether there is still milk in the fridge. Homeostasis is balance through motion. If the body gets too warm, sweat glands help cool the skin, and blood vessels near the surface can widen to release heat. If the body gets too cold, blood vessels in the skin can narrow, muscles may shiver, and metabolism can shift to preserve warmth. If microbes slip through a scratch, the immune system begins responding. If a blood vessel is injured, clotting can start. If cells become damaged or worn out, many can be repaired, replaced, recycled, or removed. The idea that the body maintains an internal balance has deep roots in physiology. In the 19th century, the French physiologist Claude Bernard described the importance of the body's internal environment. He recognized that the cells of an animal are not directly exposed to the outside world in a simple way. They live within an inner sea of fluid, blood, nutrients, salts, gases, and chemical signals. For life to continue, that inner environment must be kept stable enough for cells to do their work. Later, in the early 20th century, the American physiologist Walter Cannon developed this idea further and gave it the name homeostasis. Cannon studied the ways the body responds to stress, fear, hunger, injury, and changing conditions. His work helped show that the body is not just a collection of separate parts, but an integrated system, constantly communicating with itself to preserve internal balance. That balance is not effortless. It is maintained by endless small adjustments. Repair happens at many scales at once. At the smallest level, DNA inside cells is continually exposed to damage. DNA can be affected by copying errors when cells divide, by ultraviolet light from the sun, by chemicals from the environment, and by reactive molecules produced during normal metabolism. This does not mean the body is falling apart every second in some theatrical way. It means that life is chemically active, and chemically active things need maintenance. Cells have systems that help repair DNA. They can detect certain kinds of damage, remove faulty sections, rebuild missing pieces, and correct copying mistakes. If a cell is too badly damaged to continue safely, it may undergo apoptosis, controlled form of cell death. This sounds severe, but it is often protective. A damaged cell quietly removes itself so the surrounding tissue can remain healthier. It is not glamorous, but then again, most good maintenance work rarely is. At a larger scale, repair becomes more visible. A small cut in the skin sets off a precise sequence of events. Blood vessels react, platelets gather, a clot begins to form, immune cells move into the area, damaged material is cleared away, new tissue is built, skin cells spread across the wound, and over time the surface closes. What looks like a simple scab is actually the roof of a very busy biological construction site. Bone, too, is constantly maintained. It may feel solid and silent, but bone is living tissue filled with cells, blood vessels, nerves, collagen, and minerals. Specialized cells called osteoclasts break down old or damaged bone, while osteoblasts build new bone. This process, known as bone remodeling, helps the skeleton repair microscopic damage and adapt to the forces placed upon it. Every walk, every climb, every carried shopping bag, every ill-advised attempt to move furniture alone leaves information that the skeleton can respond to over time. The immune system also takes part in constant maintenance. Immune surveillance is the process by which immune cells move through blood, lymph, and tissues, watching for infection, injury, abnormal cells, and signs of damage. Some immune cells respond quickly and generally. Others are highly specific. Some remember previous infections. Others help clear debris and guide tissue repair. The immune system is not only a defense force, it is also a cleanup crew, a communications network, and sometimes a slightly over-enthusiastic security guard. The reason all of this is necessary is simple. The body cannot be perfectly fixed forever. Living tissue is always being exposed to the world. Skin experiences friction, sunlight, pressure, heat, cold, and countless microbes. Lungs meet air with every breath. The digestive tract handles food, acids, enzymes, and an enormous community of bacteria. Muscles and tendons absorb mechanical strain. Blood vessels endure pressure. Cells use oxygen to release energy, but oxygen chemistry can also produce reactive molecules that damage cellular structures. The same processes that make life possible also create wear. Aging adds another layer. Over time, repair systems can become less efficient. Stem cells may respond more slowly. Immune regulation can become less precise. Collagen changes. Blood vessels can stiffen. DNA damage may accumulate. Some cells enter a state called senescence, where they no longer divide but continue releasing signals that affect surrounding tissue. Aging is not simply one thing going wrong. It is the long history of repair, stress, adaptation, and imperfection gradually written into the body. And yet, the body continues with remarkable patience. Most of its repair work never reaches awareness. A person does not feel every DNA repair enzyme at work. They do not notice every old protein being dismantled. They do not sense every immune cell passing through tissue, every microscopic adjustment in blood flow, or every tiny correction in body chemistry. The work succeeds much of the time because it stays quiet. The body is never really still. Stillness is only how it appears from the outside. Beneath the skin, in the blood, bones, organs, nerves, and the small hidden spaces inside cells, life is constantly maintaining the conditions that allow it to continue. Not perfectly, not endlessly, not without cost, but carefully, persistently, and with a strange kind of biological calm. The first moments after an injury are strangely quiet from the outside. A small cut may look like a thin red line, a scrape may sting, a bruise may begin as nothing more than a dull ache beneath the skin. Inside the tissue, though, the body has already noticed that the local order has changed. Cells that were sitting calmly in their proper places have been disturbed. Some have been stretched, crushed, torn, or broken open. Tiny blood vessels may have split. Fluid may be moving where it normally should not. Molecules that usually stay tucked safely inside cells are suddenly drifting into the space around them. To the body, this is information. Damaged cells release distress signals almost immediately. They do not shout, of course, because cells are very poor at shouting and generally lack the equipment. Instead, they communicate through chemistry. Injured tissue releases molecules that tell nearby cells something has gone wrong. Some of these signals come from broken cell membranes. Others come from stressed cells that are still alive, but already changing their behavior. The damaged area becomes a small chemical beacon, calling attention to itself before the conscious mind has fully understood what happened. This is one of the first important truths about repair. Healing begins before it looks like healing. In the first seconds, nearby blood vessels respond. Many small vessels briefly narrow in a process called vasoconstriction. Vasoconstriction means that the muscular walls of blood vessels tighten, reducing the space inside them and slowing the flow of blood through the injured area. This helps limit blood loss, while the body prepares the more complicated stages of repair. It is a quick, temporary response. The body cannot simply close the blood supply for too long, because injured tissue still needs oxygen, nutrients, immune cells, and chemical messages. But for a short time, narrowing the vessel is useful. It is a little like placing a careful finger over a leak before finding the proper tools. Not elegant, perhaps, but biology is often very good at practical solutions. The blood vessel itself is not just a hollow tube. Its inner surface is lined with endothelial cells. These cells form the endothelium, a living layer that helps control blood flow, clotting, inflammation, and communication between the blood and surrounding tissues. Endothelial cells are thin and easily overlooked, but they are among the most important cells in the body. They help blood remain fluid when it should be fluid, and they help guide clotting when a vessel has been damaged. Under normal conditions, endothelial cells create a smooth inner lining that discourages platelets from sticking. This is essential because blood must usually travel freely. It cannot form clots every time it feels mildly inconvenienced. But when a vessel is torn, the deeper layers beneath the endothelium are exposed. Collagen and other structural materials that are normally hidden come into contact with circulating blood. To platelets, this is a sign that the vessel wall has been breached. Platelets are small cell fragments produced in the bone marrow from much larger cells called megakaryocytes. They are not complete cells with a nucleus, but they are extremely important. They drift through the bloodstream in huge numbers, quiet and inactive until they encounter signs of damage. When they meet an injured vessel wall, they change rapidly. They stick to the damaged surface. They change shape. They release chemical signals of their own. More platelets are attracted to the area and begin sticking to one another. This gathering of platelets forms an early plug at the site of injury. It is not yet the strong, organized clot that will come later, but it is the first physical attempt to seal the breach. This platelet plug is fast. It can begin forming within seconds. That speed matters because the body has to reduce blood loss before repair can become slower and more deliberate. A tiny wound does not need a grand construction project immediately. It needs a temporary barrier. The platelets provide that first patch, rather like a biological version of someone hurriedly putting a bucket under a leak and pretending this was the plan all along. While platelets gather, chemical messengers spread through the injured area. Histamine is one of the best known. It is released by cells such as mast cells and helps blood vessels become more leaky in nearby tissue. This may sound unhelpful at first, because leaking is not usually considered a triumph of engineering, but increased vessel permeability allows immune cells and proteins to leave the bloodstream and enter the damaged tissue, where they can begin the work of defense and cleanup. Prostaglandins are another group of chemical messengers. They're involved in pain, inflammation, blood vessel changes, and fever. In an injured area, prostaglandins help shape the local response, making the tissue more sensitive and contributing to the familiar ache or tenderness that often follows damage. Pain is not pleasant, but it is useful information. It encourages protection. It reminds the body not to keep scraping, twisting, biting, stretching, or generally annoying the injured area. Cytokines are signaling proteins used by immune and tissue cells to communicate. They help organize inflammation, guide immune cell behavior, and influence how strongly the body responds. Chemokines are a related group of signals that act rather like chemical trail markers, helping attract immune cells toward the place where they are needed. Together, cytokines and chemokines help turn a damaged patch of tissue into a coordinated repair zone. This early response is not random swelling and redness. It is an organized biological conversation. The National Institutes of Health has supported and collected a vast amount of biomedical research into inflammation, blood vessel biology, clotting, immune responses, and tissue repair. That modern understanding shows how many overlapping systems are involved even in a small injury. What looks simple from the outside is full of careful timing. Blood vessels narrow, then later widen. Platelets stick, signal, and gather. Endothelial cells sense damage and help guide the response. Chemical messengers call immune cells into position. Pain, heat, swelling, and redness begin to appear because the body is changing the local environment. A bruise follows the same general principle, though the surface may remain unbroken. Tiny blood vessels beneath the skin are damaged, allowing blood to leak into surrounding tissue. At first, the mark may look red or purple as blood collects under the skin. Over time, the colour changes as the body breaks down and clears the blood components. The bruise is not just a stain, it is evidence of cleanup and recycling beneath the surface. A scrape, a cut, and a bruise all begin differently, but each one tells the body that a boundary has been disturbed. The response begins with recognition. Something has changed. Something needs sealing. Something needs cleaning. Something needs rebuilding. The remarkable part is how quickly this begins. Before the wound looks organized, before a scab forms, before tenderness fully settles in, the body has already entered its first emergency phase. It has slowed blood loss, summoned platelets, activated chemical signals, and begun preparing tissue for the more complex work ahead. Repair does not begin with the scar. It does not even begin with the scab. It begins in those first quiet seconds, when damaged cells release their signals, blood vessels narrow, platelets gather, and the living lining of the circulation helps decide how the body should respond. The first emergency response is fast, local, and precise enough to be trusted thousands of times across an ordinary life. And once the early plug is forming, the body can begin building something stronger, a temporary seal made from blood itself. Blood has a strange double life. Most of the time it must remain fluid. It has to move easily through arteries, veins, and tiny capillaries, carrying oxygen, nutrients, hormones, immune cells, heat, and waste products from place to place. It cannot afford to thicken every time a vessel bends, or a heartbeat pushes it forward, or a person stands up too quickly and quietly regrets the decision. But when a blood vessel is damaged, blood has to do something completely different. It has to stop flowing freely at that spot. It has to become sticky, organized and solid enough to seal a breach. It has to turn from a moving river into a temporary wall. That changes blood clotting, also called coagulation. It is easy to imagine clotting as blood simply drying out, especially when a surface wound becomes a dark scab. But clotting is not just drying. It is a carefully controlled emergency repair system. Platelets gather at the damaged vessel wall. Clotting factors in the blood activate one another. Calcium helps key reactions happen. Damaged tissue provides signals that guide the process. A protein called fibrinogen is converted into fibrin, and fibrin forms a mesh that strengthens the early platelet plug. The result is not a perfect repair, it is a temporary seal. That matters. The body does not need to rebuild the whole damaged area in the first few moments. It needs to stop blood loss, reduce contamination, and create a stable surface where slower healing can begin. A clot is the body buying time. The first part of this seal often begins with platelets. These small cell fragments stick to exposed collagen and other damaged structures in the vessel wall. Once attached, they change shape, become more active, and release chemical signals that call in more platelets. They begin, clumping together into a soft plug. This platelet plug is fast, but on its own it is not strong enough for every wound. It needs reinforcement. That reinforcement comes from the clotting cascade. The clotting cascade is a chain of biochemical reactions in which inactive proteins in the blood become activated in a precise sequence. These proteins are called clotting factors. The word cascade is useful because each step helps trigger the next, like a quiet molecular line of falling dominoes, though the dominoes are floating in blood and taking their work very seriously. The goal of this cascade is to produce thrombin, an enzyme that converts fibrinogen into fibrin. Fibrinogen is soluble, meaning it can dissolve in the blood. Fibrin is different. It forms long strands that link together into a mesh. That mesh wraps through and around the platelet plug, trapping red blood cells and other material. It turns the early soft plug into a more stable clot. Fibrin is one of the great practical materials of the body. It does not look grand. It does not get the same attention as the brain or heart. But without it, even small injuries would be far more dangerous. It is biological netting, woven at the site of damage, holding the repair patch together until living tissue can begin to rebuild beneath it. Calcium is also essential in several stages of coagulation. It helps clotting factors bind to surfaces and interact correctly. Without calcium, the cascade cannot proceed normally. This is one reason blood clotting is not just one event, but a coordinated chemical process involving blood cells, proteins, minerals, vessel walls, and injured tissue. The liver is central to this system because it produces many of the clotting factors that circulate in the blood. Several of those clotting factors depend on vitamin K for their proper production. Vitamin K helps the liver make clotting proteins in a form that can bind calcium and function correctly. This is why vitamin K is scientifically important in blood coagulation, and why liver disease can sometimes affect the body's ability to clot. The clot may appear at the injury site, but some of its essential ingredients were prepared elsewhere, quietly manufactured long before the scrape, cut, or bruise ever happened. Clotting has to be powerful, but it also has to be limited. Too little clotting can cause dangerous bleeding. If the body cannot make a strong enough seal, blood loss may continue. Even small wounds can become serious when clotting is impaired. Conditions such as haemophilia show how important individual clotting factors can be. In haemophilia, certain clotting proteins are missing or reduced, making it harder for blood to form stable clots. But too much clotting creates the opposite problem. Blood that clots inside vessels when it should remain fluid can block circulation. A clot forming in the wrong place is called a thrombus. Thrombosis can reduce or stop blood flow, depriving tissues of oxygen. If a clot breaks away and travels through the bloodstream, it can become an embolus, lodging somewhere else in the body. This is why clotting is both protective and potentially dangerous. The same system that saves blood at a wound can become harmful if activated in the wrong place. The body prevents unnecessary clotting in several ways. Healthy endothelial cells help keep the inner surface of blood vessels smooth and clot-resistant. Natural anticoagulant systems limit the clotting cascade. Blood flow itself helps dilute and carry away activated clotting factors so they do not spread too far. Later, once the damaged tissue is more stable, the body can begin breaking down the clot through a process called fibrinolysis, using an enzyme called plasmin to digest fibrin. A good clot is local. It forms where needed, holds long enough to help, and then gradually gives way as repair continues. The same basic logic can look different depending on where the injury occurs. On the skin surface, a clot may dry and combine with dead cells and other material to form a scab. A scab is a protective cover over a wound. It helps reduce further bleeding, blocks some outside contamination, and shelters the new tissue forming underneath. It is not just old blood sitting there doing nothing. It is part of a temporary protective structure. A deeper internal clot is different. Inside a blood vessel or under the skin, the clot is not exposed to air in the same way, so it does not become a dry surface scab. In a bruise, for example, small vessels break beneath the skin and blood leaks into surrounding tissue. Clotting helps limit the bleeding, while immune cells and enzymes later break down and clear the trapped blood. The changing colours of a bruise show the body gradually processing hemoglobin and other blood components. It is not pretty in a glamorous sense, but it is tidy in its own slow, purple, greenish-yellow way. The 19th century physician Rudolf Virchow helped shape modern understanding of abnormal clotting. Virchow's triad describes three broad factors that increase the risk of thrombosis: changes in blood flow, injury to the vessel wall, and changes in the composition of the blood. Slow or turbulent flow can make clotting more likely. Damage to the vessel lining can expose surfaces that encourage clot formation. Changes in the blood itself can make it more prone to clotting. Virchow's triad is still useful because it shows that clotting is not only about the blood, it is about the relationship between flowing blood, living vessels, and the chemical state of the body. A clot is not just a lump, it is a response to conditions. In ordinary wound healing, clotting is one of the body's earliest acts of repair. It seals, stabilizes, and protects. It gives immune cells time to arrive, gives tissue cells a surface to work around, and gives the body a chance to move from emergency response into cleanup and rebuilding. A scab may look simple, it may look like the end of the story, but beneath it, the body is only beginning. The temporary seal has done its first job. Blood loss has slowed. The breach has been covered. Now the deeper work can continue as inflammation arrives not as a mistake, but as a controlled fire that clears the way for healing. Inflammation has a terrible reputation. It is often treated as if it were simply a problem, a biological nuisance, something swollen and angry that the body has somehow failed to prevent. But inflammation is not by itself a mistake. It is one of the body's most important repair tools. It is a controlled response to injury, infection, irritation, or damage, and when it is working properly, it helps turn a wounded area into a place where healing can begin. The familiar signs of inflammation are easy to recognize. A cut becomes red around the edges, a sprained ankle becomes warm and swollen, a bruise feels tender, a sore throat feels raw. These sensations can be uncomfortable, but they are not random. Redness often means increased blood flow. Warmth usually comes from that same local rush of blood and chemical activity. Swelling happens as fluid and immune cells move into the affected tissue. Tenderness appears because nerves become more sensitive, encouraging the injured part to be protected. In ancient and later medical traditions, inflammation was described through a set of classic signs. Rubor means redness, calla means heat, tumour means swelling, dolor means pain. Later another sign was added, functio laesa, meaning loss of function. In plain terms, an inflamed area can become red, warm, swollen, painful, and harder to use normally. A swollen finger may bend less easily. A sore knee may resist being asked to behave like nothing happened. The body is not being dramatic. It is trying to reduce further harm while repair gets organised. Inflammation begins when damaged tissue releases signals that attract immune cells and change the behaviour of nearby blood vessels. After clotting has helped seal a breach, the body still has a messy problem to solve. There may be microbes in the wound. There may be dead cells, broken fragments, leaking fluids, and damaged extracellular matrix, the supportive material that surrounds cells. The area needs cleaning, protection, and coordination. One of the first major immune cell types to arrive is the neutrophil. Neutrophils are fast responders. They circulate in the blood and can move quickly into injured or infected tissue. Their main job is to attack microbes and help contain immediate threats. They can engulf invaders, release antimicrobial substances, and contribute to the early defense of a wound. Neutrophils are useful, but they are not delicate. They are more like emergency responders with heavy boots. In the right place, at the right time, they are essential. If their activity continues too long or becomes excessive, the chemicals they release can also damage surrounding tissue. This is one reason inflammation has to be controlled. It must be strong enough to defend, but not so strong that it turns the repair site into a miniature battlefield that nobody remembered to tidy up. Macrophages arrive as another crucial part of the inflammatory response. Their name means large eaters, which is unusually direct for biology. Macrophages engulf dead cells, microbes, fragments, and debris through a process called phagocytosis. This process became central to modern immunology through the work of Elie Metchnikoff, a scientist in the late 19th and early 20th centuries who observed cells engulfing foreign material. His work helped reveal that immune defense was not only about substances floating in the blood, but also about living cells actively consuming invaders and damaged matter. Macrophages are not only cleanup crews, they're also repair coordinators. Early in an inflammatory response, macrophages can behave in a more attack-focused way, helping to destroy microbes and clear danger signals. Later, as the wound becomes cleaner, they can shift towards signals that support rebuilding. They release molecules that influence fibroblasts, blood vessel growth, collagen production, and tissue remodeling. In other words, macrophages help decide when the site should move from defense into reconstruction. That shift is vital. Inflammation is supposed to have a rhythm. It begins, intensifies, does its work, and then resolves. Resolution is not simply inflammation fading because the body gets bored. It is an active process. Chemical signals change. Immune cells stop being recruited in large numbers. Dead neutrophils are cleared away. Macrophages help calm the area. Tissue cells begin rebuilding. The body tries to restore balance. This is why short-term inflammation is protective. If a splinter breaks the skin, inflammation helps prevent infection and clear the damaged area. If a muscle is strained, inflammatory signals help start repair. If bacteria enter a wound, immune cells need to arrive quickly. Without inflammation, the body would be much less able to defend itself or heal properly. But inflammation becomes harmful when it lasts too long, happens too strongly, or appears when it is not truly needed. Chronic inflammation is different from the short-lived inflammation of an ordinary wound. It can persist for weeks, months, or years, and it can gradually damage tissue. Persistent joint inflammation, as seen in conditions such as rheumatoid arthritis, can harm cartilage and bone. Long-lasting irritation in tissues can keep immune cells active and delay normal repair. Chronic inflammation can be like a fire that was meant to clear a path, but never quite goes out. The controlled fire image is useful because fire can be protective or destructive depending on timing, location, and intensity. A small controlled burn can clear dead material and make room for new growth. An uncontrolled fire consumes what it was meant to protect. Inflammation works in a similar way. It is not good or bad in a simple sense. It is a tool, and like most powerful tools, it needs boundaries. This is also why the visible signs of inflammation are not just annoyances. Redness brings more blood to the area. Warmth reflects increased circulation and activity. Swelling carries immune cells and proteins into damaged tissue. Tenderness encourages rest. Reduced function may protect the area from further strain. None of this feels especially poetic when a toe has been stubbed on furniture in the dark, but the biology is doing its best under difficult domestic circumstances. After a wound has been sealed by a clot, inflammation helps decide what happens next. It calls in the cells that remove microbes and debris. It changes blood vessel behavior. It sends chemical instructions through the damaged tissue. It prepares the site for rebuilding by clearing what cannot remain and signaling what must be restored. The body cannot simply build new tissue on top of chaos. It has to clean, sort, defend, and organize first. Inflammation is that organizing flame. When it works well, it burns briefly and carefully, clearing the way without consuming the whole structure. Then it softens, quiets, and hands the work over to the slower patient processes of repair. Beneath the redness and swelling, the body is not merely reacting. It is deciding how to move from damage toward restoration, one chemical signal and one wandering immune cell at a time. Before the body can rebuild, it has to tidy the damage. That may sound almost too ordinary for something as complex as healing, but it is one of the most important truths in repair. New tissue cannot be laid down properly over a confused mixture of dead cells, trapped bacteria, broken fibers, leaking fluids, clotted material, and chemical debris. A wound is not simply an empty gap waiting to be filled. It is a disturbed landscape, and before construction can begin, the body has to clear the ground. This is why healing is not only about building, it is also about cleaning, sorting, recycling, and removing what no longer belongs. In medicine, the removal of damaged, dead, or contaminated tissue is called debridement. In a clinical setting, debridement may be done by trained professionals to help a serious wound heal. But the body performs its own microscopic version of this process all the time. Immune cells move into damaged tissue. Enzymes break down material that cannot be reused in its current form. Dead cells are cleared. Microbes are attacked. Fragments of broken tissue are dismantled, carried away, or prepared for recycling. It is not glamorous work. It is the biological equivalent of clearing rubble after a storm, except the rubble is made of cells, proteins, blood components, and the occasional bacterium having a very bad afternoon. Neutrophils are often among the first major cleanup and defense cells to arrive. They are fast, abundant immune cells that travel in the blood and move quickly into damaged or infected tissue. Their speed is useful because wounds can expose the body to microbes, and microbes do not wait politely while the repair crew gets organized. Neutrophils can engulf bacteria and release antimicrobial substances. They can also release enzymes and reactive molecules that help destroy invaders. In the early stages of injury, this makes them extremely valuable. They help reduce the risk that a wound becomes a doorway for infection. But neutrophils are powerful in a slightly rough way. The same chemicals that can damage microbes can also harm nearby healthy tissue if they are released for too long or in the wrong amount. This is why inflammation and cleanup must be controlled. The body needs neutrophils to arrive quickly, act strongly, and then be cleared away when their job is done. If they linger too long, the cleanup can begin to damage the room it was meant to restore. Once neutrophils have done much of the urgent early work, macrophages become increasingly important. Macrophages are immune cells with a remarkable range of duties. They can engulf bacteria, digest dead cells, remove fragments, release chemical signals, and help guide the tissue toward repair. Their name means large eaters, which is one of those rare scientific names that says exactly what it means and then quietly goes back to work. One of the key processes performed by macrophages is called efferocytosis. Efferocytosis is the clearing of dead or dying cells, especially cells that have undergone controlled self-removal. This matters because dead cells cannot simply be left scattered through damaged tissue. If they break apart in an uncontrolled way, their contents can increase inflammation and irritate the surrounding area. When macrophages clear dead cells through efferocytosis, they do more than remove waste. They help calm inflammation. The act of swallowing and processing dead cells can shift macrophages towards signals that encourage repair rather than attack. In this way, cleanup becomes a turning point. The wound begins to move away from emergency defense and toward rebuilding. This is one of the more elegant parts of healing. The body doesn't just remove debris because the debris is untidy. It removes debris because the presence or absence of that material changes the chemical mood of the wound. A site filled with broken cells and microbial signals stays inflammatory. A site where dead cells have been cleared can begin to soften into repair. Around all of this cellular activity is the extracellular matrix. The extracellular matrix is the supportive scaffold that surrounds cells. It is not just empty packing material, it helps give tissues their structure, strength, stretch, and organization. It also stores signals that influence how cells move, grow, divide, and repair. Several important materials are found in this matrix. Collagen provides strength and structure, especially in skin, tendons, ligaments, bone, and scar tissue. Elastin allows certain tissues such as skin, lungs, and blood vessels to stretch and recoil. Fibronectin helps cells attach, move, and organize during repair. Proteoglycans help hold water and influence the texture and resilience of tissues. Together, these materials form a living scaffold, more like an active landscape than a passive frame. When tissue is injured, the extracellular matrix is often damaged too. Collagen fibers may be torn. Structural proteins may be cut apart. The chemical signals stored in the matrix may be released or altered. This damaged scaffold has to be assessed and remodeled. Some parts are broken down, some are reused, some are replaced. New cells need a surface to move across and signals to guide them. Without a workable matrix, repair becomes far more difficult. Modern research is revealing just how many cell types participate in this process. Institutes such as the Wellcome Sanger Institute have contributed to large-scale efforts in cell mapping and tissue biology, helping scientists understand the different cells present in healthy and damaged tissues. This kind of work shows that healing is not carried out by one heroic cell type with a tiny cape. It is a coordinated effort involving immune cells, blood vessel cells, fibroblasts, skin cells, nerve-related cells, and many others. Cell mapping also helps explain why repair differs from one tissue to another. Skin, liver, muscle, bone, tendon, and nerve tissue each have their own cellular neighborhoods. They contain different support cells, different stem cell populations, different blood supplies, and different extracellular matrix structures. The basic logic of cleanup and rebuilding is shared, but the details vary. In a healing wound, damaged material must be removed without destroying too much healthy tissue. Microbes must be attacked without allowing inflammation to become endless. Dead cells must be cleared in a way that encourages calm. The extracellular matrix must be broken down where it is damaged and rebuilt where structure is needed. This is a careful balancing act, not a simple sweep of the biological broom. And this cleaning phase is why wounds do not instantly become new tissue. The body is deliberate because it has to be. A rushed repair built over debris would be weaker, messier, and more vulnerable to infection. The damaged area must first be made ready. It must be defended, cleared, softened, and organized. Only then can the next visible transformation begin. Once the wound is cleaner, once the inflammatory signals begin to change, and once the scaffold is prepared for new growth, skin cells can begin moving across the surface. Blood vessels can start extending into the repair zone. Fibroblasts can lay down fresh structural material. The body can shift from clearing the site to rebuilding the barrier, slowly returning the damaged surface to something whole. Skin is so familiar that it is easy to underestimate it. It is simply there, quietly wrapped around the body, meeting the world first. It feels ordinary because it is always present. Yet skin is the body's largest protective barrier, a living boundary between the inner world and everything outside it. It keeps fluids in, keeps many microbes out, helps regulate temperature, senses touch and pain, produces sweat, grows hair, and repairs itself with a patience that is easy to miss until a small wound appears. Skin is not just a thin covering. It has layers, structures, cells, vessels, nerves, glands, and tiny living systems folded into it. The outer layer is the epidermis. This is the surface barrier, built largely from cells called keratinocytes. Keratinocytes produce keratin, a tough protective protein that helps give the skin its strength and waterproofing. These cells are constantly being renewed, rising from deeper parts of the epidermis toward the surface, where older cells are eventually shed. In a very calm and unglamorous way, every person is slowly leaving tiny pieces of themselves everywhere, which is not a comforting thought for a sofa, but is excellent biology. Among the cells of the epidermis are melanocytes. Melanocytes produce melanin, the pigment that helps give skin its colour and offers some protection against ultraviolet light from the sun. Melanin does not make skin invincible, but it helps absorb and scatter some ultraviolet radiation, reducing damage to DNA in skin cells. Beneath the epidermis is the dermis. The dermis is thicker and richer in structure. It contains collagen and elastin, which help give skin strength and stretch. It contains blood vessels that bring oxygen and nutrients. It contains nerves that detect pressure, temperature, pain, itch, and gentle contact. It contains sweat glands, which help cool the body. It contains hair follicles, which produce hair and also hold important cells that can contribute to repair after certain injuries. When skin is wounded, the body has to restore this barrier as quickly and safely as possible. A break in the skin is not just a surface problem. It is an opening in one of the body's main defenses. Fluid can be lost. Microbes can enter. Underlying tissue can be exposed. So the body first seals the breach with clotting, then clears debris through inflammation and immune activity, and then begins the slow return of the barrier. One of the key processes in this return is reepithelialization. Reepithelialization means that epithelial cells, especially keratinocytes, move across the wound surface to close the gap. These cells can migrate from the wound edges and, in some cases, from structures such as hair follicles. They do not simply wait for the empty space to fill beneath them. They begin moving, spreading, and covering the wound bed, creating a new surface layer. This movement can happen beneath a scab. A scab may look like the main event because it is the part most visible from the outside, but it is only a temporary cover. Underneath it, skin cells are moving across the wound, immune cells are clearing material, and deeper tissue is being rebuilt. The scab is more like a roof over a work site than the completed building. It protects the area, but it is not the healed skin itself. This is why picking a scab can be a surprisingly effective way of annoying the body. The scab helps protect the delicate tissue underneath. If it is pulled away too early, it may reopen tiny blood vessels, disturb migrating skin cells, introduce microbes, and slow the repair that was quietly progressing below. The body can usually recover from this interruption, but it would probably prefer not to be given extra admin. While keratinocytes are helping restore the surface, fibroblasts move into the wound area below. Fibroblasts are connective tissue cells that produce collagen and other extracellular matrix materials. The extracellular matrix is the supportive scaffold around cells, and in a healing wound it becomes essential. Fibroblasts help lay down the material that gives the new tissue strength. At first, this framework is not as organized as normal skin. It is more provisional, more like temporary scaffolding than polished architecture. Collagen is especially important here. It gives strength to the healing tissue, helping the wound resist being pulled open. Over time, collagen can be remodeled, reorganized and strengthened, though healed skin often does not regain the full strength or structure it once had. Repair is remarkable, but it is not always perfect restoration. As the wound rebuilds, it also needs a renewed blood supply. This is where angiogenesis becomes important. Angiogenesis is the growth of new blood vessels from existing ones. Healing tissue needs oxygen, nutrients, immune cells, and chemical signals. New tiny vessels grow into the repair zone to support that work. This is one reason healing tissue can look pink or red and feel tender. It is not simply irritated, it is being supplied, rebuilt, and reconnected. The redness of healing skin can be a sign of increased circulation. The tenderness may come from sensitive nerves, inflammatory signals, and the vulnerability of new tissue. The area may look slightly raised, moist, or delicate as fresh cells and matrix materials settle into place. It is a temporary state, but a meaningful one. The body is not merely covering the wound. It is trying to restore a living barrier. The early study of tissues outside the body was shaped in part by the work of Alexis Carrel, a surgeon and researcher associated with early tissue culture and wound research in the 20th century. Some of his wider views and historical legacy are complicated, but his experimental work helped push scientists towards studying how tissues survive, grow, and repair under controlled conditions. Modern wound biology has moved far beyond those early studies, using cell biology, molecular signaling, imaging, genetics, and tissue engineering to understand repair in far greater detail. That modern understanding shows that even a tiny scab represents a full biological sequence. Blood clotting forms a temporary seal, inflammation helps defend and clear the area, macrophages and other immune cells remove dead material, keratinocytes migrate across the wound, fibroblasts lay down collagen, new blood vessels grow in, the extracellular matrix is rebuilt and remodeled, nerves may become sensitive as the region recovers. A small wound, then, is not small to the cells living through it. It is a local rebuilding project with barriers to restore, signals to interpret, structures to replace, and timing to manage. From the outside, it may be just a scab on a finger. From the inside, it is a carefully coordinated return to separation between the body and the world. And when that return is complete, the surface may close smoothly, or it may leave behind something more permanent, a mark, a ridge, a pale line, a small reminder that repair does not always mean returning exactly to what was there before. A scar is not a sign that the body failed. It is a sign that the body made a choice. Not a conscious choice, of course. There is no tiny project manager in the skin wearing a hard hat and deciding that this wound will receive the practical but slightly uneven finish. But in biological terms, scarring is a compromise. The body often prioritizes closing a wound quickly and strongly over recreating the original tissue perfectly. In a world full of microbes, friction, sunlight, movement, and the occasional sharp corner of furniture, speed can matter more than elegance. Perfect restoration would be ideal. The skin would return exactly as it was, with the same texture, strength, colour, sweat glands, hair follicles, nerves, and flexible structure. But for many injuries, especially deeper ones, the body does not rebuild the original architecture in every detail. It patches the breach, it restores a barrier, it reinforces the area. It chooses survival over flawless design, which is deeply relatable, if not always cosmetically satisfying. The main material in many scars is collagen. Collagen is the most abundant protein in the human body, and it provides strength and structure in skin, tendons, ligaments, bone, blood vessels, and many other tissues. In normal skin, collagen fibers are arranged in a more organized basket-like pattern that helps the skin stretch, resist tension, and move with the body. During wound repair, fibroblasts enter the damaged area and begin producing new collagen and other extracellular matrix materials. This early collagen is often laid down quickly. It can be disorganized, dense, and uneven compared with the surrounding skin. That is not because the body's being careless. It is because the first priority is to stabilize the wound. A fast repair does not always have time to restore the original microscopic pattern. Later, the scar enters a remodeling phase. Some collagen is broken down. New collagen is laid down. Fibers become more aligned along lines of tension. The scar may flatten, soften, and fade over months or even years. Early scar tissue may look red or raised because blood vessels and active repair cells are still present. Over time, as remodeling continues, it may become paler and less noticeable. But even remodeled scar tissue is usually not identical to undamaged skin. Fibroblasts are central to this process. They are the cells that build much of the wound's structural framework. They produce collagen, fibronectin, and other extracellular matrix components, helping fill and strengthen the damaged region. Some fibroblasts can become myofibroblasts. Myofibroblasts are specialized repair cells that have contractile features, a little like smooth muscle cells. They help pull the edges of a wound closer together, reducing the open gap that must be filled. This contraction can be useful. In a healing wound, drawing the edges inward helps close the space and reduce the amount of new tissue needed. But like many repair processes, it must be controlled. Too much contraction can restrict movement, especially after larger burns or deep injuries. The body's repair systems are powerful, but power is not the same thing as perfection. Scar tissue is often different because it lacks many of the structures found in normal skin. Hair follicles may not return in the scarred area. Sweat glands may be absent. The surface texture may be smoother, shinier, raised, sunken, or tighter. The arrangement of collagen may reduce flexibility. Nerves may regrow imperfectly, which can make scars numb, sensitive, itchy, or oddly aware of themselves at inconvenient times. This is why a scar can remain as a visible memory of injury. The body has restored coverage and strength, but it has not recreated the full living complexity of the original skin. A scar is not simply new skin. It is a repair patch made from living tissue, functional enough to protect, but different enough to be recognized. Sometimes the body builds too much scar tissue. A hypertrophic scar is a raised scar that stays within the boundaries of the original wound. It forms when collagen production is excessive during healing. creating a thickened area that may improve over time. A keloid goes further. It grows beyond the edges of the original wound, spreading into nearby skin. Keloids can be firm, raised, itchy, painful, or simply persistent, and they show how the repair system can overbuild. This overbuilding is not random laziness from the body, as tempting as it might be to blame it for poor interior design. It reflects the complexity of signaling during wound repair. Fibroblasts, immune cells, growth factors, mechanical tension, genetics, and local inflammation all influence how much collagen is produced and how it is remodeled. In some people and in some areas of the body, the balance tilts toward too much scar formation. So scarring can mean too little restoration, too much rebuilding, or simply a practical middle ground. The wound closes, the barrier returns. The tissue becomes stronger than it was during the open injury, though not always as flexible or complete as before. This raises a larger and more mysterious question. Why do humans scar so often, while some animals regenerate missing parts with far greater precision? The study of regeneration has a long scientific history. Thomas Hunt Morgan, better known for his work in genetics using fruit flies, also studied regeneration in animals such as planarians. Planarians are small flatworms with extraordinary regenerative abilities. Under the right conditions, pieces of a planarian can regrow missing structures, revealing a very different biological strategy from ordinary human wound repair. Other animals show striking regenerative powers too. Salamanders can regrow limbs. Zebrafish can repair parts of the heart. Some species can rebuild tissues in ways that mammals usually cannot. These animals do not simply patch wounds with scar tissue. They can, in certain contexts, recreate complex structures. Humans do have some regenerative abilities. Skin renews. Blood cells are constantly replaced. The liver can regrow after partial loss. Bone can heal impressively well. The lining of the intestine renews rapidly. But when it comes to many complex tissues, especially deep skin injuries, major nerve damage, heart muscle after a heart attack or lost limbs, human repair is far more limited. Scarring may be part of the reason. A fast scar closes a wound and reduces infection risk, which is useful for survival. But that same quick closure can also create a fibrous barrier that prevents the more elaborate rebuilding seen in regenerating animals. In evolutionary terms, a scar may not be beautiful, but it can be good enough to keep an organism alive. And good enough is a powerful phrase in biology. The body is not designed to produce perfection. It is shaped by survival, reproduction, energy limits, injury risk, infection pressure, and the trade-offs of living in a physical world. A scar is one of those trade-offs made visible. It says that the body repaired the breach, restored the barrier, and chose a strong patch over a perfect reconstruction. A scar can be pale, raised, smooth, tight, tender, or almost invisible. It can mark a childhood fall, a surgery, a burn, a scrape, an accident, or a moment hardly remembered. But beneath its surface is a story of clotting, inflammation, immune cells, fibroblasts, collagen, blood vessels, and remodeling. It is not failure written on the skin, it is survival written in collagen. A scar is a kind of biological compromise. It closes a wound, restores a barrier, and gives tissue enough strength to carry on. But it also raises a very natural question. If the body can repair so much, why can it not simply rebuild everything exactly as it was? Why can skin scar instead of returning perfectly? Why can a broken bone heal, while a lost finger cannot usually grow back? Why does the human body patch so many injuries, while some animals seem to perform repairs that look almost like quiet magic? In nature, true regeneration does exist. It is just not spread evenly across life. Salamanders are famous for their ability to regrow limbs. If a salamander loses part of a leg, it can, under the right conditions, rebuild bone, muscle, nerves, blood vessels, skin, and connective tissue in the correct arrangement. Axolotls, a type of salamander often used in regeneration research, are especially remarkable. They can regrow limbs, repair parts of the heart, restore sections of the spinal cord, and heal certain wounds with far less scarring than mammals. Planarians are even stranger. These small flatworms can regenerate astonishing amounts of their bodies. Their abilities come from a population of adult stem cells called neoblasts, which can give rise to many different cell types. A small piece of a planarian can, in some circumstances, rebuild missing structures with an accuracy that makes human wound healing look rather like patching a wall with a tea towel and hoping guests do not notice. Zebrafish also have powerful regenerative abilities. They can repair damaged fins, parts of the heart, and some nervous system tissues. In regeneration biology, zebrafish are important because they are vertebrates, like humans, yet they can restore certain structures far more effectively than mammals can. This makes them useful for studying the genetic and cellular signals that might explain why some animals regenerate while others mainly scar. A key structure in limb regeneration is the blastema. A blastema is a mass of cells that forms near the site of injury in animals capable of strong regeneration. These cells are not just random repair material. They can contribute to rebuilding complex structures. In a regenerating salamander limb, cells near the injury can enter a more flexible state, multiply, and then help form the correct tissues in the correct positions. Bone must become bone. Muscle must become muscle. Nerves must reconnect. Skin must cover the surface. The new limb must not merely fill space. It has to become organized. That level of organization is one of the great challenges of regeneration. Rebuilding tissue is not like pouring plaster into a crack. A limb is a three-dimensional structure with pattern, direction, joints, nerves, blood supply, and different tissue types placed in very specific relationships. Regeneration requires not only growth, but guidance. Cells need to know where they are, what they should become, and when to stop. Humans do not lack repair. Humans repair constantly. But most human repair is limited compared with the dramatic regeneration seen in salamanders, axolotls, planarians, and zebrafish. One reason mammals often scar instead of regenerating may involve the immune system. Mammalian immune responses are powerful, fast, and highly developed for defending against infection. That is useful, especially for warm-blooded animals with complex tissues and active lives. But a strong inflammatory response can also encourage scar formation. Rapid sealing protects the body from blood loss and microbes, but it may reduce the opportunity for slower, more complete regeneration. Tissue complexity also matters. Human limbs are large, mechanically demanding structures with bones, tendons, ligaments, blood vessels, nerves, muscles, skin, and specialized sensory systems. Rebuilding all of that would require extraordinary coordination. The body would need to restart developmental programs in an adult tissue environment without accidentally creating disorder. Cancer risk is another possible trade-off. Regeneration requires cells to divide, move, change identity, and respond to growth signals. These are useful abilities when controlled, but dangerous when uncontrolled. Mammals have strong systems to prevent cells from dividing inappropriately, because uncontrolled cell growth can become cancer. A body that is very good at stopping dangerous growth may also be less able to restart the flexible growth programs needed for full regeneration. Evolution does not always favor the most impressive repair. It favors what works well enough for survival and reproduction in a particular environment. Fast wound closure may have been more valuable for many mammals than slow, perfect rebuilding. A scarred wound that closes quickly can keep infection out and blood in, It may not be elegant, but biology is not above a practical shortcut. Evolution has produced many wonders, but it has also produced the knee, so perfection was clearly not the only design goal. Still, humans do regenerate in limited ways. The liver is one of the most famous examples. After partial removal or injury, the liver can regrow a significant amount of its mass, largely through the division of remaining liver cells. This is not the same as regrowing a lost limb, because the liver is restoring tissue mass rather than reconstructing an intricate external shape with bones and joints. But it's still one of the body's most impressive repair abilities. Skin renews continually. New cells form in deeper layers of the epidermis and move upward as older cells are shed. Blood is constantly renewed from stem cells in the bone marrow. Red blood cells, white blood cells, and platelets are produced throughout life. Bone can heal fractures with remarkable strength, forming temporary repair tissue and then remodeling it over time. The lining of the intestine also renews rapidly, because it faces constant wear from digestion, microbes, and chemical exposure. These examples show that the human body is not poor at renewal, it is selective. Some tissues replace themselves often, some repair strongly after damage, others have very limited recovery. Heart muscle, many parts of the nervous system, and complex lost structures such as limbs do not regenerate well in humans. This is where stem cells become important. Stem cells are cells that can renew themselves and produce specialized cell types. They are not all the same. Adult stem cells exist in tissues such as bone marrow, skin, intestine, and muscle, where they help maintain and repair particular parts of the body. They're usually limited in what they can become. A blood-forming stem cell in bone marrow produces blood cells, not a new kneecap, however useful that might occasionally be. Embryonic stem cells is different. They come from early embryos and can give rise to almost any cell type in the body. This broad potential makes them scientifically powerful, but also ethically complex. Their use is carefully regulated and remains a subject of serious discussion. Induced pluripotent stem cells change the field in a different way. In 2006, the Japanese scientist Shinya Yamanaka and his colleagues showed that mature cells could be reprogrammed into a stem cell-like state by introducing specific factors. These induced pluripotent stem cells are important because they suggested that cell identity is not as permanently fixed as once thought. A mature cell could, under the right laboratory conditions, be pushed back toward a more flexible state. Yamanaka's work transformed regenerative medicine. It opened new possibilities for studying disease, testing drugs, modeling development, and perhaps one day creating replacement tissues. It did not mean that doctors could suddenly regrow human limbs on request, preferably by Tuesday. Biology remains far more complicated than that, but it showed that the instructions inside cells can sometimes be reset in profound ways. The difference between scarring and regeneration is therefore not a simple matter of the body knowing or not knowing how to heal. It is a question of timing, immune response, cell flexibility, tissue patterning, cancer protection, evolutionary history, and the local environment around the injury. Humans are not built for dramatic whole-part regeneration. They are built for constant maintenance, rapid sealing, selective renewal, and survival through repair that is often good enough rather than perfect. A scar may seem less impressive than a regrown limb, but in its own way it tells the same deeper story. Living bodies are always negotiating between restoration and safety, between rebuilding and control, between what would be ideal and what can be done quickly enough to keep life going. And within that negotiation, stem cells remain some of the body's most important reserves, waiting quietly in specific tissues, helping renew blood, skin, muscle, intestine, and more, not as magic, but as carefully regulated sources of future repair. Stem cells are often spoken about as if they are tiny miracle workers. The reality is more interesting and much more careful. Stem cells are not magic repair dust sprinkled through the body whenever something goes wrong. They are living cells with particular abilities, in particular places, responding to particular signals. Some can renew themselves over long periods. Some can produce specialized cell types. Some help maintain tissues that are constantly wearing out. Others remain quiet until injury, strain, or normal turnover calls them into action. In the adult human body, stem cells act as specialized repair reservoirs. They are found in tissues such as bone marrow, skin, intestine, and skeletal muscle, where they help support renewal and replacement. They do not usually wander freely around the body looking for problems to solve, like microscopic tradespeople with suspiciously small vans. Most adult stem cells live in specific environments called niches. A stem cell niche is the local neighborhood that helps control the cell's behavior. It provides signals that tell the stem cell when to stay quiet, when to divide, and what kinds of cells it should produce. This control is essential. A repair reservoir is only useful if it is regulated. Too little activity, and tissues cannot renew properly. Too much activity, and the risk of disordered growth can increase. The body needs stem cells to be flexible, but not reckless. One of the clearest examples is found in bone marrow. Bone marrow contains hematopoietic stem cells. Hematopoietic means blood forming, and these cells give rise to the major cell types in the blood. From this source come red blood cells, white blood cells, and platelets. Red blood cells carry oxygen from the lungs to tissues throughout the body. They are packed with hemoglobin, the protein that binds oxygen and gives blood its red colour. Red blood cells do not last forever, so they must be replaced continuously. White blood cells are central to immune defense, helping the body recognize infection, respond to injury, remember past threats, and clear damaged material. Platelets are small cell fragments involved in clotting, helping seal damaged blood vessels when bleeding begins. All of this depends on the bone marrow's ability to keep producing new blood cells. Every day, the body makes enormous numbers of them. This is not dramatic healing in the visible sense. It is maintenance on a grand scale, hidden inside bones. The blood moving through the body is not a fixed supply. It is constantly refreshed. The intestine offers another striking example of rapid renewal. The lining of the gut faces a very demanding life. It must absorb nutrients, manage digestive enzymes and bile acids, interact with trillions of microbes, and protect the body from harmful substances. It is exposed to chemical stress, mechanical wear, and microbial activity every day. Unsurprisingly, the cells lining the intestine are replaced frequently. Intestinal stem cells sit in small gland-like structures called crypts. From these crypts, new cells are produced and move into the lining, replacing older cells that are shed. This renewal helps maintain the barrier between the contents of the gut and the internal tissues of the body. It also allows the intestine to recover from ordinary wear and certain forms of injury. The speed of intestinal renewal is one reason the gut is so biologically impressive. It is a working border, a chemical processing surface, a microbial ecosystem, and a repair site all at once. It is also a reminder that some of the body's most important maintenance happens in places that do not look especially poetic from the outside. Biology is not always glamorous, but it is rarely lazy. Skin also depends on stem cells and renewing cell populations. Cells in deeper layers of the epidermis help produce new keratinocytes, which move upward as older surface cells are shed. Hair follicles contain stem cell populations that can contribute to hair growth, and in some cases help with skin repair after injury. This is one reason skin can maintain itself so effectively across everyday scratches, dryness, friction, and exposure. Skeletal muscle has its own repair reservoir. Muscle fibers are long, specialized cells built for contraction, and they can be damaged by exercise, strain, or injury. Alongside muscle fibers are satellite cells, a type of adult stem cell important for muscle repair. Satellite cells usually sit quietly near muscle fibers. When muscle is damaged or stressed, they can become activated, divide, and help repair existing fibers or contribute to new muscle material. This is part of what happens after exercise. During unfamiliar or demanding activity, tiny disruptions can occur in muscle tissue. The body responds with inflammation, repair signaling, protein synthesis, and satellite cell activity. Over time, with enough rest and nutrition, muscle can adapt. It may become stronger, better coordinated, or more resistant to the same stress in future. This does not mean soreness is always a sign of good repair, or that more damage always means more improvement. The body prefers sensible loading, not heroic chaos. Muscle adaptation depends on repeated signals, recovery, adequate energy, and time. Even cells appreciate a reasonable schedule. Stem cells, then, are part of the body's spare capacity. They allow certain tissues to keep renewing and repairing themselves, but their abilities are specific and limited. A hematopoietic stem cell makes blood cells. An intestinal stem cell supports the gut lining. A satellite cell helps skeletal muscle. These cells are powerful because they are suited to their tissue, not because they can do absolutely anything anywhere. This distinction matters because stem cell science is sometimes exaggerated. Legitimate stem cell research is one of the most important areas of modern biology and medicine. It has transformed understanding of development, tissue repair, blood diseases, immune disorders, organ models, and regenerative medicine. Hematopoietic stem cell transplantation, often known as bone marrow transplantation, is an established treatment for some blood cancers and serious blood or immune conditions. But not every advertised stem cell treatment is scientifically proven. Some commercial claims suggest that stem cells can easily cure a long list of unrelated conditions, reverse aging, rebuild joints, repair nerves, or generally persuade the body to behave as if it has read a motivational poster. In reality, safe and effective therapies require careful evidence, proper clinical trials, quality control, regulation, and long-term monitoring. Cells are living treatments, and living treatments can behave unpredictably. The International Society for Stem Cell Research provides guidance on responsible stem cell research and clinical translation. It warns against unproven interventions and encourages clear evidence, ethical oversight, and patient safety. That cautious approach is not there to make the science less exciting. It exists because the science is powerful enough to require care. The best way to understand stem cells is to see them neither as fantasy nor disappointment. They are not magical cure-all cells. They are not empty hype either. They are regulated sources of renewal shaped by tissue context, local signals, age, injury, and the body's need for balance. They help make blood, renew gut lining, support skin, and repair muscle. They reveal that the body carries hidden reserves, but those reserves are organized, constrained, and deeply specific. The body's repair potential is real. It is just not unlimited, and few tissues show that quiet potential more elegantly than bone, a structure that seems solid and inert from the outside yet can sense stress, rebuild after fracture, and remodel itself across a lifetime. Bone looks like the body's quiet architecture. It gives shape, support, leverage, protection, and the useful ability to stand up without becoming a soft heap of opinions. It can seem like inert scaffolding, a hard internal frame that simply holds everything in place. But bone is not dead stone. It is living tissue, filled with blood vessels, nerves, collagen, minerals, marrow, and active cells that build, break down, sense, repair, and reshape it throughout life. The hardness of bone comes largely from minerals, especially calcium phosphate, arranged in a form called hydroxyapatite. These minerals give bone its stiffness and strength under compression. But bone is not just mineral, it also contains collagen, a flexible structural protein that helps it resist bending and cracking. Mineral gives bone its stone-like firmness. Collagen gives it a little toughness. Together they make bone strong without making it as brittle as a breadstick with ambitions. Inside many bones is marrow, where blood cells are made. Blood vessels pass through bone, bringing oxygen and nutrients. Nerves allow bone to sense pain and injury. And within the hard matrix live specialized cells that maintain the tissue. Bone is not a fixed object. It is a changing, responsive organ. 3 cell types are especially important. Osteoblasts are bone-building cells. They produce new bone matrix and help mineralize it. Osteoclasts are bone-resorbing cells. They break down old or damaged bone, releasing minerals back into circulation and clearing space for renewal. Osteocytes are mature bone cells embedded within the bone matrix. They help sense mechanical strain and communicate with other bone cells, guiding where bone should be strengthened, maintained, or removed. This constant process is called bone remodeling. It happens even without obvious injury. Tiny areas of older bone are broken down and replaced. Microscopic damage from ordinary movement is repaired. The skeleton adjusts to changing forces, activity, and age. It is a slow conversation between load and structure. When a bone breaks, this living nature becomes especially clear. A fracture is not simply a crack in a dry object. It is an injury to a vascular, cellular tissue. Blood vessels tear. Bone cells are damaged. The surrounding soft tissues may also be bruised or disrupted. The body responds with a sequence that is surprisingly elegant. First, a blood clot forms around the fracture. This clot, sometimes called a fracture hematoma, helps fill the gap and begins the repair environment. It contains blood cells, inflammatory signals, and proteins that attract immune and repair cells. As with the skin wound, clotting is not the final repair. It is the first temporary stabilizing step. Then inflammation begins. Immune cells arrive to clear dead cells, fragments, and debris. Blood vessels in the area become more active. Chemical signals help recruit cells that will take part in rebuilding. This early stage can involve swelling, tenderness, and pain, which are not signs that the body has forgotten how to behave, but signs that the repair site is being organized. Next comes the soft callus. A callus in this context is not the thickened skin on a hand or foot. It is a temporary bridge of repair tissue forming around and between the broken bone ends. The soft callus contains collagen and cartilage-like material. It helps stabilize the fracture, though it is not yet hard enough to restore full strength. Over time, the soft callus is replaced by a hard callus. Osteoblasts lay down new bone, and the tissue becomes mineralized. The fracture becomes much more stable. This new bone may initially be bulky and somewhat disorganized, because the body's priority is strength and continuity. It is a little like building a sturdy temporary bridge before worrying about making it elegant. The final stage is long-term remodeling. Osteoclasts remove excess or poorly arranged bone, while osteoblasts build new bone in a more organized pattern. The shape gradually becomes closer to the original structure. This process can continue for months or even years depending on the fracture, age, health, blood supply, and the forces placed on the bone. Bone often heals more completely than many soft tissues because it can rebuild mineralized living structure and remodel it over time. A well-aligned fracture with good blood supply can sometimes regain impressive strength and shape. But healing is not automatic or guaranteed. Alignment matters. If the broken ends are not positioned properly, The repair may form at an angle or with poor function. Blood supply matters because repair cells need oxygen and nutrients. Age matters because healing often slows later in life. Nutrition matters, especially protein, vitamin D, calcium, and other materials needed for tissue repair. Movement matters too because the bone needs enough stability to heal, but also receives useful signals from carefully managed loading. Too much movement too early can disrupt repair. Too little loading for too long can weaken bone. Anyone who has worn a cast may have noticed that the protected limb can feel thinner or weaker afterward. That is because bone and muscle both respond to use. The body is practical. It tends not to maintain expensive structure without reason. Biology is not wasteful. It is just occasionally a bit ruthless with the maintenance budget. This responsiveness is captured by Wolff's law. Wolff's law is the principle that bone adapts to the loads placed upon it. The 19th century German surgeon Julius Wolff helped describe this relationship between mechanical stress and bone structure, and his ideas became important in orthopedic science. In broad terms, stronger or repeated loading can encourage stronger bone architecture, while reduced loading can contribute to bone loss. This is why weight-bearing exercise can help maintain bone strength. Walking, resistance training, and other activities that place controlled stress on the skeleton give bone cells signals that the structure is needed. It is also why astronauts can lose bone density in microgravity, where the skeleton is not loaded in the same way. The bone is still alive, still remodeling, but the signals have changed. Wolff's law does not mean every stress is beneficial. Bones need appropriate loading, recovery, nutrition, and healthy hormonal signaling. A gentle walk and a sensible strengthening routine are very different from repeatedly asking a tired bone to do something unreasonable until it files a formal complaint in the form of a stress fracture. Still, the broader principle is beautiful. Bone listens. It records the forces of a life in its structure. A tennis player's serving arm, a runner's legs, a dancer's feet, a casted wrist, a healing fracture, all show that bone is responsive tissue, shaped by use and protected by repair. So bone is not merely the body's stone framework, it is living stone. If stone could bleed, feel pain, make blood cells, rebuild itself, and quietly judge posture from the inside. It breaks, clots, inflames, bridges, hardens, and remodels. It can restore itself with a completeness many tissues cannot match, but only through careful coordination between cells, blood supply, minerals, collagen, mechanical signals, and time. And that same idea of repair through use, strain, rest, and rebuilding appears in another familiar part of the body, where damage is often microscopic, soreness is common, and adaptation can make tissue stronger than before. Muscle repair is not only something that happens after an obvious injury. It happens after ordinary use. It happens after a long walk, an unfamiliar workout, carrying heavy bags, climbing stairs, lifting suitcase, or spending an afternoon doing some mysterious household task that somehow uses muscles no one remembered owning. Skeletal muscle is built for movement, force, and adaptation, and every time it is challenged, it receives information about what the body might need from it in future. Muscle is made from long fibers that contract to produce movement. These fibers are bundled together and surrounded by connective tissue, blood vessels, and nerves. When a muscle works, especially during intense or unfamiliar activity, tiny disruptions can occur in the muscle fibers and the surrounding connective tissue. These are sometimes casually called microtears, though the word can make the process sound more dramatic than it usually is. Most of the time, this is not serious damage. It is part of the normal relationship between strain, repair, and adaptation. The body responds to this disruption with a careful repair process. Chemical signals are released. blood flow increases, immune cells help clear damaged material. Muscle cells begin rebuilding proteins. Support cells become more active. The tissue does not simply return to its previous state in every case. If the challenge is repeated sensibly, the muscle may adapt so that it can handle similar work more easily in future. This is where muscle hypertrophy comes in. Muscle hypertrophy means an increase in the size of muscle fibers. It is one of the main ways skeletal muscle adapts to repeated loading, especially resistance training. The process is driven by mechanical tension, repair signaling, nutrition, hormones, and repeated training over time. The muscle senses that it is being asked to produce force. In response, it increases the machinery that helps it do that work. This does not happen instantly. A single workout does not transform a muscle into a heroic sculpture overnight, no matter what certain fitness adverts may whisper at vulnerable people. Muscle adaptation takes repeated signals and enough recovery. The body needs the stimulus of work, but also the materials and time to respond. Protein synthesis is central to this repair and growth. Protein synthesis is the building of new proteins inside cells. Muscle fibers contain proteins that allow contraction, such as actin and myosin, as well as many structural and regulatory proteins. After exercise or injury, the balance between protein breakdown and protein building becomes important. If the body has enough nutrients, rest, and appropriate signaling, it can repair damaged proteins and build new ones. Blood flow also matters. Muscles need oxygen and nutrients to function and recover. Blood brings amino acids, glucose, fatty acids, hormones, immune cells, and other materials into the tissue. It also helps remove waste products. During and after activity, circulation changes to meet the needs of working muscle. This is one reason movement can feel warming. The tissue is not just doing mechanical work, it is becoming chemically busy. Among the most important repair cells in skeletal muscle are satellite cells. Satellite cells are muscle-associated adult stem cells that sit near muscle fibers, usually in a quiet state. When muscle is damaged by strain, exercise, or injury, satellite cells can become activated. They can divide, and some of their descendants can fuse with existing muscle fibers. This helps donate additional nuclei to the fibers, supporting repair and growth. This is important because skeletal muscle fibers are unusually large cells with many nuclei. A single nucleus can only support a certain region of cellular activity. By adding nuclei, satellite cells help the muscle fiber manage the demands of repair and adaptation. They are not magic cells floating around with a universal toolkit, but they are vital local helpers, quietly waiting beside the tissue they serve. Muscle recovery is also linked to inflammation, though again, inflammation is not automatically bad. After demanding exercise, local inflammatory signals can help coordinate repair. Immune cells clear damaged material and release signals that influence rebuilding. The goal is not to erase inflammation completely, but to let the right amount happen at the right time, then settle. Like many repair systems, it is useful when it arrives, does its job, and leaves before becoming annoying. Delayed onset muscle soreness is one familiar result of this process. It often appears after unfamiliar or intense exercise, especially movements that lengthen a muscle while it is under tension, such as walking downhill, lowering a weight, or discovering that squats are apparently a personal betrayal. The soreness usually develops hours after the activity and may peak over the following day or two. Delayed onset muscle soreness is linked to microscopic disruption, inflammation, chemical sensitivity, and tissue adaptation. It is not simply caused by lactic acid sitting in the muscle. That older explanation is too simple. Lactate is produced during intense activity, but it is cleared fairly quickly, and is also used as a fuel and signaling molecule. The lingering soreness that appears later is better understood as part of the repair and adaptation response to unfamiliar stress. Rest is essential because repair needs time. During recovery, the muscle rebuilds proteins, restores energy stores, adjusts cellular machinery, and resolves inflammation. Sleep, nutrition, hydration, and sensible spacing between hard efforts all help. Without enough recovery, repeated strain can outpace repair. With too much rest and no loading at all, muscle can shrink and weaken. The body listens carefully to what is asked of it, and sometimes takes silence as a budget cut. Gradual loading is one of the most important principles in muscle adaptation. The muscle needs enough challenge to stimulate change, but not so much that damage overwhelms repair. This is why training programs often increase difficulty slowly. A muscle adapts best when the signal is repeated, manageable, and followed by recovery. Repair is not just fixing what was damaged, it is preparation for the next demand. Exercise physiology researchers, including Roger Fielding and others studying muscle adaptation and aging, have helped clarify how muscle responds to resistance training, nutrition, inactivity, and the passage of time. This research is especially important because muscle mass and strength tend to decline with age, a process often called sarcopenia. Resistance exercise and adequate protein intake can help support muscle function in many people, though individual needs vary. The broader lesson is that muscle is responsive tissue. It changes with use, disuse, injury, recovery, age, hormones, nutrition, and blood supply. It is not merely a set of ropes pulling on bones. It is a living system that senses load, repairs microscopic disruption, and adapts to repeated effort. This makes muscle repair different from the healing of a cut, but not entirely separate from it. There are still signals, immune cells, proteins, blood vessels, stem-like support cells, and remodeling. The scale is different. The purpose is different. But the underlying theme remains the same. The body is always trying to preserve function. After effort, the muscle does not simply complain, though sometimes it does that too, very convincingly. It rebuilds, it replaces damaged proteins, it adjusts its structure, it prepares itself for what may come next. And yet not all movement tissues heal with the same ease. Some are strong precisely because they are dense and tough, but the toughness comes with slower repair. The tendons and ligaments that connect movement together show another side of healing, where strength, blood supply, collagen, and patience become especially important. Tendons and ligaments are built for strength. They are not soft, dramatic tissues that announce themselves every moment. Most of the time they work in the background, transferring force, stabilizing joints, guiding movement, and allowing muscles and bones to act together. A step, a reach, a jump, a turn of the head, a gentle stretch in the morning, all depend on these quiet connective structures doing their jobs without complaint. Tendons connect muscle to bone. When a muscle contracts, the tendon helps transmit that force to the skeleton, producing movement. The Achilles tendon, for example, connects the calf muscles to the heel bone and helps with walking, running, and pushing off the ground. Ligaments connect bone to bone. They help stabilize joints, keeping bones aligned while still allowing controlled movement. The ligaments of the ankle, knee, shoulder, wrist, and spine are all part of the body's system of restraint and guidance. Both tendons and ligaments are rich in collagen. Collagen fibers give them tensile strength, meaning they are good at resisting pulling forces. In a healthy tendon, collagen fibers are arranged in a highly organized direction, lined up with the forces the tendon usually experiences. This careful alignment is one reason tendons can transmit powerful muscle forces without tearing during ordinary movement, Ligaments also rely on collagen, though their arrangement varies depending on the joint and the directions of force they must resist. But the same qualities that make tendons and ligaments strong can also make them slow to heal. Many tendons and ligaments have a limited blood supply compared with more vascular tissues such as muscle or skin. Blood brings oxygen, nutrients, immune cells, signaling molecules, and repair materials. When circulation is modest, the repair process tends to move more slowly. Fewer supplies arrive quickly, and waste products may be cleared less efficiently. This does not mean tendons and ligaments cannot heal. It means they often heal with patience, gradual remodeling, and a pace that can feel deeply unimpressed by human impatience. An ankle sprain is one familiar example. A sprain happens when a ligament is stretched or torn, often after a joint twists beyond its usual range. The injured ligament may swell, hurt, and feel unstable. The body responds with inflammation, clotting in tiny damaged vessels, immune activity, and later collagen production. But restoring the ligament's strength and coordination takes time. The tissue has to rebuild not only continuity, but also organized structure. Achilles tendon problems show another side of connective tissue repair. The Achilles tendon carries large forces during walking, running, jumping, and climbing. Repeated overload, sudden strain, age-related changes, or poor recovery can contribute to pain and degeneration in the tendon. In many tendon problems, the issue is not a simple clean tear that heals in a neat line. It can involve disorganized collagen, altered cell behavior, swelling within the tendon, and changes in the surrounding matrix. Rotator cuff tendon injuries are also common, especially around the shoulder. The rotator cuff is a group of muscles and tendons that help stabilize and move the shoulder joint. These tendons can become irritated, partially torn or fully torn, especially with age, repeated overhead movement, trauma, or long-term wear. Because the shoulder is so mobile, and because some regions of these tendons have limited blood supply, healing can be slow and sometimes incomplete. A key idea in tendon and ligament recovery is collagen alignment. When repair begins, fibroblasts and related connective tissue cells produce new collagen. Early repair collagen may be laid down in a less organized pattern. It helps reconnect the tissue, but it may not immediately match the smooth, parallel structure of a healthy tendon or the carefully arranged fibers of a ligament. Over time, mechanical signals help guide remodeling. Fibers can become more aligned with the direction of force. The tissue can become stronger and more functional. This is why controlled movement can sometimes support better repair than complete inactivity. The word controlled is important. Too much force too soon can disrupt healing tissue, but too little loading for too long can also be unhelpful, because tendons and ligaments need mechanical signals to remodel properly. They are designed to respond to tension. Gentle, progressive loading can encourage collagen fibers to organize along useful lines, helping the tissue regain strength. This principle does not mean that every injury should be moved immediately or aggressively. Severe tears, fractures, surgical repairs, or unstable joints may need protection. The right amount of movement depends on the tissue, the injury, the stage of healing, and medical guidance. But broadly, connective tissue is not passive string. It is living tissue that senses load and adapts to it. The scientist Keith Barr has contributed important work in connective tissue adaptation and tendon biology, including research on how tendons and ligaments respond to mechanical loading, and how collagen synthesis is influenced by activity and recovery. This kind of research connects laboratory biology with practical ideas. Tissues need signals, nutrients, rest, and time. Strength is not rebuilt by wishing. It is remodeled through carefully repeated demand. Scar tissue can restore continuity in tendons and ligaments, but it may not perfectly restore original movement. The new tissue may be stiffer, less elastic, or less smoothly organised than the original. A repaired ligament may hold the joint together but still leave some stiffness or instability. A tendon may regain strength but remain sensitive, thickened, or less efficient at gliding. The body can reconnect and reinforce, but it does not always recreate the exact architecture that existed before injury. This is one reason recovery from connective tissue injuries can feel frustrating. Pain may fade before the tissue has fully regained strength. Swelling may reduce while remodeling is still underway. A person may feel almost normal, then discover that the tendon or ligament is still not ready for sudden twisting, sprinting, lifting, or heroic gardening. Connective tissue has its own calendar, and it is not always interested in being convenient. Still, its repair is impressive in its own way. Tendons and ligaments are dense, strong, and highly specialized. Their healing requires collagen production, inflammation control, blood supply, mechanical signaling, gradual loading, and long-term remodeling. The body has to rebuild not just material, but direction. Strength depends on structure, and structure depends on how fibers are arranged. This adds an important nuance to the story of repair. The same body can heal skin relatively quickly, remodel bone powerfully, adapt muscle after exercise, and yet take far longer to restore a tendon or ligament. Healing is not one speed or one style. It changes with each tissue structure, job, blood supply, and cellular environment. A tendon is not a muscle. A ligament is not skin. Bone is not a scab. Each tissue repairs according to what it is and what it must do. And behind all of these processes, another system keeps moving through the body, defending, cleaning, communicating, and sometimes deciding whether repair will settle calmly or remain inflamed. The immune system is not separate from healing. It is woven into almost every step, from the first emergency response to the slow return of strength. The immune system is often imagined as a defense force. That image is not wrong. The immune system does protect the body from bacteria, viruses, fungi, parasites, and other threats. It recognizes danger, attacks invaders, remembers past infections, and helps keep the inner world of the body from being overrun by things that would very much like to move in rent-free. But the immune system is not only a battlefield, it is also a repair crew, a communication network, a cleaning service, and a regulator of healing. When tissue is damaged, immune cells help decide what happens next. They remove microbes, clear dead cells, release chemical signals, influence blood vessels, guide fibroblasts, shape inflammation, and help damaged areas return towards stability. Without the immune system, the body would not simply be more vulnerable to infection, it would also be much worse at organizing repair. The immune system has two broad arms: innate immunity and adaptive immunity. Innate immunity is the fast general response. It is always ready, because it does not need to learn the exact identity of every threat before acting. It includes the physical barriers of the body, such as skin and mucous membranes, which keep many microbes out before deeper defense is needed. It includes inflammation, the controlled response that brings blood flow, immune cells, and signaling molecules to damaged or infected tissue. Neutrophils are part of innate immunity. They arrive quickly at sites of infection or injury, engulf microbes, and release substances that help destroy invaders. They are fast and useful, though not always gentle. Macrophages are also part of innate immunity. They swallow dead cells, microbes, and debris, but they also release signals that influence whether tissue remains inflamed or begins rebuilding. A macrophage can behave like a cleaner, a guard, a messenger, and a repair coordinator, which is quite a lot of responsibility for something without a calendar. Dendritic cells are another important part of the immune system. They help collect information from tissues, and present pieces of microbes or damaged material to cells of the adaptive immune system. In this way, they act as a bridge between the fast general response and the slower, more specific response. Natural killer cells are also part of innate immunity. They can recognize and destroy certain infected or abnormal cells, especially when those cells show signs that something inside them is wrong. Complement proteins add another layer. The complement system is a group of proteins in the blood and tissue fluids that can help mark microbes for destruction, attract immune cells, and in some cases punch holes in microbial membranes. It is one of those systems that sounds almost medieval when described plainly, but it is a normal part of immune defense. Adaptive immunity is more specific. It takes longer to develop during a first encounter, but it can recognize particular targets with remarkable precision. B cells can produce antibodies, which are proteins that bind to specific parts of microbes or toxins. Antibodies can block invaders, mark them for destruction, and help immune cells recognize what needs removing. T cells have several roles. Some T cells help coordinate immune responses. Others can kill infected cells directly. The adaptive immune system also creates immune memory. After certain infections or vaccinations, the body can respond more quickly and strongly if it meets the same threat again. This is one of the great biological advantages of adaptive immunity. It learns, remembers, and improves its response over time. In repair, adaptive immunity can be helpful, but it must be carefully managed. A strong immune response is useful when a wound is contaminated with bacteria. It is useful when viruses infect cells. It is useful when abnormal cells need to be removed. But immune activity is powerful chemistry carried out by living cells, and powerful chemistry in living tissue is never something to leave entirely unsupervised. The body must regulate immune responses because defense can damage the very tissues it is meant to protect. Neutrophils can release enzymes and reactive molecules that harm nearby cells. Inflammation can cause swelling, pain, and tissue stress. T cells and antibodies can be highly effective, but if they target the wrong thing, they can attack the body itself. The immune system has to recognize danger without treating every human cell as suspicious simply because it happens to be nearby. This ability to avoid attacking the body's own healthy tissues is called immune tolerance. Immune tolerance is one of the central ideas in immunology. It allows the immune system to respond to harmful invaders while leaving most self tissues alone. This distinction between self and non-self was shaped in modern immunology by researchers including Peter Medawar and Frank McFarlane Burnett, whose work helped explain how the immune system can learn tolerance and why that tolerance matters. When immune tolerance fails, autoimmunity can occur. Autoimmunity means the immune system mistakenly targets the body's own tissues. In rheumatoid arthritis, immune activity contributes to inflammation in joints. In type 1 diabetes, immune attack damages insulin-producing cells in the pancreas. In multiple sclerosis, immune processes damage myelin, the protective covering around nerve fibers in the central nervous system. These conditions are very different from one another, but they show the same broad principle. An immune system that loses tolerance can turn defense into damage. This is why repair depends not only on activating the immune system, but also on calming it down again. A wound needs immune cells to prevent infection and clear dead material. But if the inflammatory response keeps going too long, healing can stall. Chronic inflammation can interfere with tissue rebuilding, encourage excessive scarring, and keep cells in a state of chemical alarm. A repair site needs protection, but it also needs peace. In healthy healing, the immune system changes over time. The early response is more defensive. It focuses on sealing, recognizing damage, attacking microbes and clearing debris. Later, the signals shift. Macrophages help clear dead neutrophils. Inflammatory chemicals decrease, repair signals increase, blood vessels grow, fibroblasts produce extracellular matrix. Tissue cells begin rebuilding the damaged area. The immune system does not leave the scene, it changes its role. This is what makes immunity so deeply woven into repair. It is not a separate department waiting until infection appears. It is present in the first moments after injury. It helps decide whether inflammation resolves or becomes chronic. It influences scarring. It shapes the environment in which stem cells, fibroblasts, blood vessels, and epithelial cells do their work. The immune system is therefore not just the body's army, it is also its restoration service. It clears broken material, checks for danger, sends instructions, guides rebuilding, and tries to restore balance without causing too much collateral damage. When it works well, the result is almost invisible. A cut closes, a bruise fades, a sore throat passes, a strained muscle settles. The body returns quietly toward ordinary life, and beneath even this immune activity, repair continues at a smaller scale still. Inside individual cells, damage is being detected, proteins are being folded or dismantled, DNA is being corrected, and tiny internal systems are working to keep each living unit stable enough to survive. Every visible repair begins with smaller repairs that cannot be seen. A cut closing on the skin, a bruise fading, a muscle recovering after strain, all depend on work happening deep inside individual cells. Each cell is a living unit, but it is not a simple little bag of fluid. It contains membranes, proteins, organelles, genetic instructions, energy systems, waste processing machinery, and signaling networks. It has jobs to do, but it must also maintain itself while doing them. That is one of the quiet difficulties of being alive. A cell cannot stop all activity, close the shutters, and spend the afternoon on repairs. It has to keep functioning while also fixing damage. It must make proteins, respond to signals, manage its energy, move materials, protect its DNA, maintain its membrane, and decide when a problem can be corrected or when the safest option is to remove itself entirely. The cell is not still. It is a tiny working world, and like any working world, it produces wear. DNA is one of the most important things a cell must protect. DNA carries the instructions for making proteins and guiding cellular function. It is copied when cells divide, read when genes are used, and stored in a chemical environment that is constantly active. Damage to DNA is not rare in the sense of being strange. It is part of normal life. Some DNA damage happens during replication, when a cell copies its genetic material before division. Copying is highly accurate but not perfect. Occasionally, the wrong chemical letter may be placed into the new strand. Some damage comes from ultraviolet light from the sun, which can alter DNA in skin cells. Some comes from reactive oxygen species, chemically reactive molecules produced during normal metabolism. Environmental exposures, certain chemicals and radiation can also damage DNA. This does not mean the body is in constant catastrophe. Cells are not helpless libraries with the roof leaking every second. They have repair systems. These systems detect many kinds of damage and correct them before they become serious. One repair pathway is base excision repair. This handles small chemical changes to individual DNA bases. If one base is damaged, specialized enzymes can recognize the problem, remove the damaged part, and allow the correct piece to be restored. It's a little like finding one smudged word in a long instruction manual and carefully replacing it, rather than throwing away the whole book. Another pathway is nucleotide excision repair. This is useful for bulkier distortions in the DNA structure, including some damage caused by ultraviolet light. Instead of removing only one damaged base, the cell cuts out a short stretch of damaged DNA. and fills in the missing section using the undamaged strand as a guide. Mismatch repair deals with mistakes that slip through during DNA copying. If the wrong base is paired with another, mismatch repair helps identify and correct the copying error. This pathway is important because dividing cells need to pass on accurate genetic instructions. Without mismatch repair, errors would accumulate more quickly. Double-strand break repair deals with one of the more serious forms of DNA damage, where both strands of the DNA molecule are broken. Cells can repair these breaks in different ways. Some methods use a matching DNA template to guide accurate repair. Others join the broken ends more directly, which can be faster but less precise. The cell's choice depends on timing, cell type, and available information. The importance of these repair pathways was clarified by scientists including Aziz Sankar, Thomas Lindahl, and Paul Modrich. Their work helped explain how cells preserve genetic information despite constant chemical stress and copying errors. Their discoveries showed that DNA stability is not simply because DNA is perfectly protected, but because living cells actively inspect, correct, and maintain it. DNA is not the only thing that needs attention. Proteins also require quality control. Proteins are the working molecules of cells. They form structures, carry signals, speed up reactions, move materials, and perform countless other tasks. But proteins must be folded into the right shapes to work properly. Heat, chemical stress, genetic errors, age, and ordinary cellular activity can cause proteins to misfold or become damaged. Chaperone proteins help with this problem. They assist other proteins in folding correctly or refolding after stress. The name is wonderfully fitting. A chaperone protein does not perform the final job for the other protein, but helps it avoid embarrassing structural mistakes on the way there. If a protein cannot be repaired, the cell can dismantle it. One major system for this is the ubiquitin proteasome system. Ubiquitin is a small protein that can be attached to damaged or unwanted proteins, marking them for destruction. The proteasome is a molecular machine that breaks tagged proteins into smaller pieces, which can then be reused. This is cellular recycling with strict quality control. Cells also use autophagy. Autophagy means self-eating, though it is less alarming than it sounds. Through autophagy, cells can package damaged components, old organelles, or unwanted material into compartments that are delivered to lysosomes, where they are broken down. This helps cells recycle building blocks and clear internal clutter. During stress or nutrient shortage, autophagy can become especially important. Organelles need maintenance too. Mitochondria, the energy-producing organelles, can become damaged and may be removed through a specialized form of autophagy called mitophagy. Membranes have to be repaired when they are disturbed. The endoplasmic reticulum, involved in protein folding and processing, has stress responses that help restore balance when too many unfolded proteins accumulate. All of this shows that repair inside the cell is not one process. It is a whole collection of monitoring, correction, recycling, and removal systems. The cell is constantly asking a practical question. Can this be fixed? Should it be recycled? Or is the damage too great? Sometimes the answer is that the cell itself should be removed. Apoptosis is programmed cell death. It is a controlled self-removal process, not a messy collapse. During apoptosis, a cell carefully dismantles itself. Its contents are packaged in a way that limits inflammation, and nearby cells or immune cells can clear the remains. This can protect the body when a cell is infected, badly damaged, no longer needed, or at risk of becoming dangerous. Apoptosis is essential during development, immune regulation, and tissue maintenance. It is also important in cancer prevention, because a cell with severe DNA damage may be safer dead than alive and dividing. This sounds harsh, but at cellular scale it is a form of protection. One damaged cell can be sacrificed so the larger tissue remains stable. There is something quietly profound about this. The body repairs itself not only by building new material, but by deciding what cannot safely remain. A protein may be refolded or destroyed. A mitochondrion may be repaired or recycled. A strand of DNA may be corrected. A cell may recover, pause, or remove itself. Repair is not always restoration. Sometimes it is editing. Inside every tissue, these microscopic decisions continue. Skin cells repair DNA after sunlight exposure. Immune cells recycle damaged components after battle. Muscle cells rebuild proteins after strain. Intestinal cells maintain themselves in a harsh chemical environment. Bone cells respond to stress and signaling. Each visible act of healing depends on countless invisible acts of cellular maintenance, and none of this work is free. Repair requires energy, oxygen, nutrients, enzymes, and time. Cells can only rebuild if they have the fuel to do it and the machinery to turn that fuel into usable power. The body's repair systems may seem quiet, but beneath that quiet is a deep energetic cost, paid constantly by the living chemistry inside every cell. Healing may look quiet from the outside, but it is expensive work. A wound closing, a bone rebuilding, a muscle recovering, or a cell repairing its DNA all require energy. The body cannot repair itself through good intentions alone, though that would be convenient and probably very popular. Cells have to move, divide, build proteins, produce collagen, pump ions across membranes, send chemical signals, maintain temperature, control blood flow, and keep ordinary life running at the same time. Repair is not a pause in normal biology. It is normal biology with extra tasks added. When skin begins to close over a wound, keratinocytes need energy to crawl across the surface. Fibroblasts need energy to make collagen and other extracellular matrix materials. Immune cells need energy to move, engulf debris and release signals. Blood vessel cells need energy to grow new capillaries. Bone cells need energy to build and mineralize new tissue. Even inflammation, which can seem like a simple swelling or redness, is metabolically active. It requires fuel, oxygen, proteins, and constant communication. The main usable energy currency of the cell is adenosine triphosphate, often shortened to ATP. For voiceover, it is best understood as adenosine triphosphate first, then ATP as the familiar acronym. ATP is not energy itself in some vague glowing sense. It is a molecule that cells use to transfer energy into useful work. When cells need to build, move, transport, signal, or repair, ATP is often involved somewhere in the process. Much of this ATP is produced with the help of mitochondria. Mitochondria are organelles inside cells, often described as powerhouses, though they are more like controlled energy conversion systems than tiny generators wearing hard hats. They help extract usable energy from fuel molecules, especially products of carbohydrates and fats, using oxygen in a highly organized series of reactions. One of the main processes involved is oxidative phosphorylation. The phrase sounds heavy, but the basic idea can be kept gentle. Cells break down fuel molecules from food into smaller chemical forms. Electrons from those fuel molecules are passed through a chain of proteins in the inner membrane of the mitochondrion. As this happens, the mitochondrion creates a difference in charge particles across that membrane. The cell then uses that difference to help produce ATP. In simpler terms, mitochondria use oxygen and fuel to help release energy in a controlled way. This is crucial because cells cannot simply burn fuel all at once like a flame. They need to capture energy gradually and safely in a form that can power life. A campfire releases heat and light very quickly. A cell needs something more controlled, more precise, and much less likely to set the furniture on fire. Oxygen is therefore essential for many repair processes. Tissues need oxygen to make enough ATP for rebuilding. This is one reason blood supply matters so much in healing. A well-supplied tissue can receive oxygen, nutrients, immune cells, and repair materials. A poorly supplied tissue may heal more slowly. Tendons, ligaments, and some areas of cartilage are examples where limited circulation can make repair difficult. Skin, muscle, and bone often have better access to blood, though even there, healing depends on the quality of circulation. But oxygen also comes with a complication. In the course of normal metabolism, cells can produce reactive oxygen species. These are chemically reactive molecules that contain oxygen. At low or controlled levels, reactive oxygen species can help with signaling. They can influence inflammation, blood vessel behavior, and cellular responses to stress. They are not automatically villains. The problem comes when reactive oxygen species are produced in excess or not properly controlled. Then they can damage proteins, lipids, DNA, and other cellular structures. This is called oxidative stress. It is one of the reasons the body needs antioxidant systems, DNA repair pathways, protein quality control, and careful metabolic balance. Oxygen helps power repair, but oxygen chemistry can also create damage that repair systems must manage. Biology often has this slightly ironic habit of making the solution part of the problem. Nutrition matters because repair requires raw materials as well as energy. Protein provides amino acids, the building blocks used to make many tissue proteins, enzymes, immune molecules, and structural materials. Collagen, for example, is a protein, and it depends on a steady supply of amino acids. Vitamin C is important for collagen synthesis. It helps enzymes modify collagen so the fibers can form properly. The Hungarian biochemist Albert Szent-Györgyi played a major role in vitamin C research, helping identify and understand this essential nutrient. A severe lack of vitamin C can cause scurvy, a disease in which collagen production is impaired, leading to weak connective tissue, bleeding gums, poor wound healing, and other problems. That is a dramatic example, but it shows how deeply repair depends on chemistry. Vitamin D and calcium matter for bone health. Calcium is a major mineral component of bone, while vitamin D helps regulate calcium absorption and bone metabolism. Iron is needed for hemoglobin, the oxygen-carrying protein in red blood cells, so it supports oxygen delivery to tissues. Zinc plays roles in immune function, wound healing, DNA synthesis, and enzyme activity. None of these nutrients acts alone, and none should be treated as magic repair powder, but together they show that healing requires materials. The body also has to balance its resources. During major injury, infection, surgery, burns, or serious illness, the demand for energy and protein can rise. The immune system becomes active. Tissue rebuilding increases. Temperature may rise during fever. The body may break down stored energy and shift metabolism to support survival and repair. In smaller everyday healing, these changes are much less dramatic, but the same principle remains. Repair has a cost. Even maintaining normal body temperature takes energy. So does keeping the heart beating, blood circulating, lungs moving, kidneys filtering, and nerves signaling. Repair has to happen on top of all that. The body is never working from an empty schedule. It is always fitting healing into a day already crowded with breathing, thinking, digesting, moving, and occasionally wondering why it entered a room. This makes healing a practical negotiation. Cells need energy, oxygen, and raw materials. They need blood flow and chemical signals. They need inflammation to begin and resolve. They need damaged material cleared and new material built. They need mitochondria to keep producing ATP, but they also need systems to manage oxidative stress. Healing is not free, but when the conditions are good enough, the body pays the cost quietly. And one of the most important conditions for recovery is rest. Not because the body becomes inactive during rest, but because rest gives repair systems a better chance to work. Sleep, especially, is not an empty state. It is a deeply active biological rhythm, one that helps regulate immunity, hormones, metabolism, memory, and the quiet maintenance of the body itself. Sleep can look like the body has finally stopped. The eyes close, muscles relax, breathing settles into a slower rhythm. The room grows quiet and the person inside it seems to have gently stepped away from the world. From the outside, sleep can appear almost like absence, a soft nightly disappearance. But sleep is not absence. Sleep is an active biological state. It supports immune function, hormone regulation, tissue repair, memory processing, metabolic balance, and the quiet maintenance of the nervous system. The body is not simply switched off. It is reorganizing its priorities. Some systems slow down. Others become more active. The brain changes its electrical rhythms. Hormones rise and fall. Immune signals shift. Energy use is adjusted. The sleeping body is not idle. It is doing work that waking life does not easily allow. This is one reason sleep and recovery are so closely linked. During sleep, especially deep sleep, the body releases growth hormone in pulses. Growth hormone is involved in tissue maintenance, muscle repair, bone metabolism, and the regulation of how the body uses fat, sugar, and protein. It does not act alone, and it is not a magic healing fluid, which is slightly disappointing but scientifically important. It is part of a larger network of hormones and signals that help coordinate growth, repair, and restoration. Deep sleep is especially associated with physical recovery. During this stage, heart rate and breathing can slow, and the body settles into a state that supports rebuilding. Muscles recovering from strain need protein synthesis, blood flow, and cellular repair. Bones require ongoing remodeling. Skin and connective tissues need collagen production and immune coordination. Sleep does not perform these tasks by itself, but it helps create a biological environment in which these processes can proceed more efficiently. The immune system is also shaped by sleep. Sleep helps regulate inflammatory signals and supports the balance between defense and calm. After infection or injury, the body often increases the desire to sleep. This is not laziness, it is part of a coordinated response. Rest allows energy to be directed toward immune activity, fever regulation, tissue repair, and recovery. Poor sleep can disrupt this balance. When sleep is short, irregular, or repeatedly disturbed, immune regulation can become less precise. Inflammatory markers may rise. Responses to infection can be weakened. Wound healing and tissue repair may be affected, especially when poor sleep is combined with stress, illness, poor nutrition, or existing health conditions. The body can still repair under imperfect conditions because it is impressively stubborn, but it may have to work with fewer advantages. Sleep also affects metabolism. During normal sleep, the body regulates glucose handling, appetite hormones, stress hormones, and energy balance. Disrupted sleep can interfere with insulin sensitivity and appetite regulation. This matters because repair depends on stable energy management. Cells need fuel and raw materials, but they also need the body's internal chemistry to remain organized enough for rebuilding. The brain has its own nighttime maintenance systems. One of the most intriguing is the glymphatic system, a waste clearance pathway associated with sleep. Research by Micah Nedegard and her research group helped bring attention to this system, showing how fluid movement in the brain may help clear metabolic waste products. The brain is a busy organ, and busy organs produce waste. During sleep, changes in brain activity and fluid movement appear to support this clearance process. The glymphatic system should not be exaggerated into a simple nightly brain rinse, as though the skull contains a tiny dishwasher with a very strict cycle. But it is an important example of how sleep supports maintenance. The sleeping brain is not merely dreaming or resting, it is regulating, sorting, clearing, and preparing itself for future waking life. Sleep also supports memory processing. Experiences from the day are not simply stored like files dropped into a cabinet. The brain strengthens some connections, weakens others, integrates new information, and reshapes memory networks. This is not separate from repair in the broad sense. A living body must maintain not only tissue, but function. For the brain, that includes memory, learning, emotional regulation, and the ability to wake into a world that still makes some kind of sense. Underlying sleep is the circadian rhythm, the body's roughly daily timing system. Circadian rhythm helps coordinate sleep and waking, hormone release, body temperature, digestion, metabolism, and many other processes. The central clock in the brain is a small region called the suprachiasmatic nucleus, located in the hypothalamus. It receives information about light from the eyes and uses that information to help synchronize the body's timing with the outside world. Light is one of the strongest signals for this system. Morning light helps set the clock. Darkness helps allow the evening rise of melatonin, a hormone associated with the timing of sleep. Many tissues also have their own local clocks, including the liver, muscles, immune cells, and fat tissue. The central clock helps coordinate them, rather like a conductor trying to keep a very large orchestra from all playing different songs at breakfast. This timing matters for repair. Immune activity, hormone release, body temperature, and metabolism all change across the day and night. A wound, a muscle, or a recovering immune system does not exist outside time. The body's internal rhythms influence when certain processes are more active and how well different systems cooperate. Still, sleep should not be treated as a miracle cure. Sleep does not instantly mend every injury, erase inflammation, rebuild cartilage, reverse aging, or politely ask a virus to leave. Healing still depends on the kind of damage, the tissue involved, blood supply, nutrition, age, overall health, medical care when needed, and many other factors. But sleep gives the body favorable conditions. It supports regulation. It helps coordinate hormones. It shapes immune balance. It gives tissues time away from some waking demands. It helps the brain process memory and clear waste. It allows the body to perform maintenance with fewer interruptions from movement, attention, stress, light, food, noise, and the endless small negotiations of being awake. So sleep is not the opposite of repair. It is one of repair's quiet allies. While the sleeping person feels absent from the world, the body continues its careful work. Cells rebuild proteins, hormones pulse through the blood, immune signals rise and settle, the brain sorts and clears, muscles recover, bones remodel, metabolism shifts. The whole organism enters a rhythm that has been shaped by evolution across countless nights. And yet, across a lifetime, these repair systems change, sleep changes, hormones change, immune responses shift, stem cells become less responsive, tissues become slower to renew. The body continues repairing, but the pace and precision of that repair gradually alter with age. Aging is not simply the body breaking. It is more subtle than that, and more interesting. Aging is partly the gradual accumulation of damage, partly the changing capacity of repair systems, and partly the long-term result of living cells trying to maintain themselves in a world that never stops applying pressure. It is not caused by one single mechanism. There is no one hidden switch that flips from youth to age, No tiny internal calendar suddenly declaring that the warranty has expired. Instead, aging comes from many processes interacting over time. DNA damage can accumulate, proteins can misfold, mitochondria can become less efficient, stem cells may become less responsive, immune regulation can change, collagen can stiffen, blood vessels can lose some flexibility. Repair remains active, but the speed, accuracy, and resilience of repair can gradually shift. The body is still working, it is just working with a longer history. One important idea in aging biology is cellular senescence. Cellular senescence is a state in which a cell stops dividing but remains metabolically active. At first, this can be protective. If a cell has accumulated damage or receives signals that something is wrong, stopping division can reduce the risk that it will become cancerous. In that sense, senescence can act like a safety brake. But senescent cells do not always sit quietly. Some release chemical signals that affect nearby tissue. These signals can attract immune cells, influence inflammation, change the extracellular matrix, and alter the behavior of neighboring cells. When senescent cells are cleared efficiently, they may be part of normal repair and protection. When they accumulate, they can contribute to chronic inflammation and tissue dysfunction. This is one of aging's recurring themes. A process that is useful in the short term can become troublesome when it persists. The idea that cells have limits to division was shaped by the work of Leonard Hayflick. In the 1960s, Hayflick showed that many normal human cells grown in laboratory culture do not divide forever. They divide a limited number of times before entering a state of growth arrest. This became known as the Hayflick limit, and it helped change the idea that ordinary cells were naturally immortal outside the body. One reason cell division is limited involves telomeres. Telomeres are protective caps at the ends of chromosomes. They help prevent chromosome ends from being mistaken for broken DNA. Each time many types of cells divide, telomeres tend to shorten. When they become too short, the cell may stop dividing or enter senescence. Telomeres are important, but they do not explain aging by themselves. It would be misleading to treat them as the whole story. Aging is not just a matter of telomeres wearing down, and longer telomeres are not automatically better in every context. Cells also need protection against uncontrolled division, which is why telomere biology is closely connected to cancer biology as well as aging. Elizabeth Blackburn, Carol Grieder, and Jack Sostak made major discoveries about telomeres and telomerase, the enzyme that can help maintain telomere length in certain cells. Their work revealed a crucial part of how chromosomes are protected and how some cells maintain their ability to divide. Telomerase is active in stem cells, reproductive cells, and many cancer cells, which again shows the delicate balance between renewal and risk. Another important concept is inflammaging. Inflammaging means the low-grade chronic inflammation associated with aging. It is not the same as the sharp inflammation around a fresh wound. It is quieter, broader, and longer lasting. It can arise from immune system changes, senescent cells, accumulated cellular stress, changes in the gut microbiome, persistent infections, metabolic shifts, and damaged molecules that keep the immune system mildly activated. This matters because repair depends on inflammation being properly timed. A short inflammatory response can defend tissue and prepare it for rebuilding, but long-term low-level inflammation can interfere with healing, damage tissues, and increase the risk of disease. Older tissues may already exist in a more inflamed background state, so a new injury can occur in an environment that is less calm and less efficient than it once was. Repair also changes because stem cells change. Adult stem cells do not disappear all at once, but their behavior can shift with age. Some become less able to divide, some respond less strongly to repair signals. Their surrounding niches can change too. A stem cell's local environment helps decide what it does, so if the tissue around it becomes inflamed, stiffened, poorly supplied with blood, or chemically altered, the stem cell may not function as well. Blood supply is another factor. Good repair depends on oxygen, nutrients, immune cells, and waste removal. As blood vessels age, some can become less flexible, and circulation in certain tissues may become less efficient. Reduced blood supply can slow the delivery of repair materials and limit the growth of new tissue. The extracellular matrix also changes. Collagen can become stiffer and more cross-linked over time, which can affect the flexibility of skin, blood vessels, tendons, and other tissues. Stiffer tissue can change how cells behave, because cells sense the physical environment around them. A young tissue and an older tissue may send very different mechanical signals, even before any obvious injury occurs. The immune system changes too. Some immune responses may become weaker, while others become more inflammatory. The body may respond less effectively to new infections, clear damaged cells more slowly, or remain inflamed for longer after injury. This does not mean older bodies stop repairing. They continue repairing every day. But the repair may be slower, less complete, or more likely to leave stiffness, scarring, or lingering irritation. Aging is therefore not a simple decline from perfect to broken. The young body is not perfect, and the older body is not merely damaged. Both are dynamic. Both are adapting. Both are repairing. But over decades, small imperfections can accumulate faster than they are resolved. Repair systems continue, yet they carry the burden of previous repairs, past, inflammation, altered tissues, and cells that have lived through years of chemical stress. There is no need to turn this into gloom. Aging is also evidence of persistence. A body that ages has survived countless infections, injuries, stresses, meals, nights, movements, memories, and ordinary days. It has patched, cleared, rebuilt, and adjusted again and again. The lines in skin, the slowing of recovery, the stiffness after rest, the fading speed of repair, all reflect not just loss, but history. The science of aging helps explain why repair changes over a lifetime. It does not reduce a person to worn parts. It shows how deeply alive the body remains, still negotiating balance, still responding to damage, still trying to maintain order, even as the work becomes harder. And because repair has limits, scientists continue to ask whether those limits can be understood, supported, and sometimes carefully extended through regenerative medicine, tissue engineering, and the next generation of biomedical research. The future of repairing the human body begins with a careful kind of hope. Not the noisy kind that promises instant replacement organs, effortless healing, or a convenient new spine by next Thursday. The real future is slower, more cautious, and far more interesting. It is being built from a better understanding of cells, signals, materials, genes, immune responses, and the environments that allow tissues to heal. This field is often called regenerative medicine. Its aim is to understand, support, or improve the body's ability to repair itself. It brings together stem cell biology, tissue engineering, biomaterials, gene therapy, organoids, and controlled immune modulation. The goal is not always to make the body do something completely unnatural. Often, it is to help the body do what it already tries to do, but more effectively, more safely, or in situations where natural repair is limited. Tissue engineering is one of the clearest examples. In tissue engineering, researchers try to combine cells, scaffolds, and signaling molecules to create conditions where tissue can grow, organize, or repair. A scaffold is a supportive structure that gives cells somewhere to attach and arrange themselves. It may be made from natural materials, synthetic materials, or a combination of both. Ideally, the scaffold gives cells guidance without getting in their way, rather like a temporary framework around a building under restoration. Cells are the living workers in this system. They may be stem cells, specialized tissue cells, or cells encouraged to behave in particular ways. Signaling molecules help tell those cells what to do. They can influence growth, movement, survival, inflammation, and differentiation, which is the process by which cells become more specialized. The challenge is not simply to grow cells. Cells can grow in the wrong pattern, the wrong place, or the wrong amount. The harder problem is organization. A tissue is not just a pile of cells. Skin has layers. Bone has mineral structure. Tendon has aligned collagen fibers. Liver tissue has blood flow and metabolic organization. Nerves need precise connections. Heart muscle must beat in coordination. To repair complex tissue, scientists have to think about shape, timing, mechanics, oxygen supply, immune response, and the signals that guide cells into the right arrangement. Biomaterials are central to this work. A biomaterial is a material designed to interact with living tissue. Some biomaterials are used in implants, wound dressings, surgical meshes, bone substitutes, or drug delivery systems. In repair science, a good biomaterial may help guide cells, reduce unwanted inflammation, degrade safely over time, or deliver signals at the right pace. The body is not easily fooled by materials placed inside it. It inspects them, coats them with proteins, sends immune cells, and decides whether to accept, isolate, inflame, or integrate them. Biology is polite only when it feels like it. Organoids are another important tool. Organoids are miniature, simplified tissue models grown in laboratories from stem cells or tissue cells. They can develop some features of organs such as the intestine, brain, liver, kidney, or lung. They are not complete organs. A brain organoid is not a tiny thinking brain. An intestinal organoid is not a miniature digestive system asking for lunch. But organoids can help scientists study development, disease, genetic conditions, infection, drug responses, and tissue behavior in ways that are difficult to observe directly inside the human body. Organoids are useful because they allow researchers to study living human-like tissue in a controlled setting. They can show how cells organize, how mutations affect development, how drugs influence tissue, and how disease processes unfold. They may also help move medicine toward more personalized testing, where cells from a particular patient could be used to study how that person's tissue responds to treatments. Gene therapy and gene editing add another layer. Some diseases and repair problems are influenced by faulty genes or harmful genetic instructions. Gene therapy aims to add, replace, silence, or modify genetic material in cells to treat disease. Gene editing goes further by changing DNA at specific locations. One major gene editing system is called clustered regularly interspaced short palindromic repeats, often paired with Cas enzymes. This system was adapted from a natural bacterial defense mechanism. In research and medicine, it can be used to guide a Cas enzyme to a chosen DNA sequence, where the enzyme can cut or alter the genetic material. Jennifer Doudna and Emmanuel Charpentier were central figures in developing this technology into a powerful gene editing tool. The promise is real, but it must be handled carefully. Editing genes inside living cells raises questions of accuracy, unintended effects, delivery, immune response, ethics, and long-term safety. A tool that can change DNA is powerful precisely because it can matter so deeply. In medicine, power without caution is not progress. It is just trouble in a clean lab coat. Controlled immune modulation is another important part of future repair. Since the immune system can support healing or interfere with it, researchers are studying ways to guide immune responses more precisely. This might mean reducing harmful chronic inflammation, encouraging helpful macrophage behavior, improving transplant acceptance, or preventing immune rejection of engineered tissues. Repair is not only about adding cells or materials, it is about creating the right environment. Major research institutions are involved in these questions around the world. The Francis Crick Institute supports advanced biomedical research into cell biology, genetics, development, disease, and tissue function. The National Institutes of Health funds and conducts a vast range of medical research, including work on regenerative medicine, immune biology, genetics, and wound healing. The European Molecular Biology Laboratory contributes to molecular biology, developmental biology, imaging, genomics, and the study of living systems at fine detail. These institutions, among many others, help move repair science from broad hope toward tested understanding. The challenges are serious. Safety comes first. Cells that grow too much can increase cancer risk. Transplanted or engineered tissue may be rejected by the immune system. New tissue needs blood supply because cells cannot survive long without oxygen and nutrients. Nerves may need to reconnect. Mechanical strength must match the job of the tissue. Ethical questions matter, especially around embryos, genetic editing, consent, access, and fairness. Regulation is essential because promising ideas must be tested before they become treatments. There is also a wide gap between laboratory promise and proven clinical care. Many discoveries work beautifully in cells, organoids, or animals, but become much harder in human bodies. Human tissues are larger, older, more varied, and more complicated than laboratory models. A treatment must not only work once in ideal conditions, it must work reliably, safely, and meaningfully for real people. Still, the direction is remarkable. Science is learning not only how the body repairs itself, but why it sometimes cannot. It is learning how cells receive instructions, how scaffolds shape growth, how the immune system guides healing, how genes influence repair, and how tissues might be supported when natural healing is not enough. The future of repair is unlikely to be one grand breakthrough that solves everything. It will more likely be many careful advances, each helping a specific tissue, disease, injury, or process. Better wound dressings. More precise immune treatments. Safer gene therapies. Improved stem cell methods. Smarter biomaterials. More accurate organoid models. Stronger understanding of aging tissue and chronic inflammation. Not magic, not science fiction pretending to be medicine, but a patient extension of what the body has been doing all along, sensing damage, sending signals, clearing debris, rebuilding structure, and trying, in its quiet biological way, to keep life going. The human body repairs itself without one single miracle system. There's no hidden master switch, no perfect internal mechanic, no one graceful process that explains everything. Instead, the body survives through countless overlapping systems working at different scales. Blood seals wounds, inflammation clears and coordinates, skin restores the barrier, scars form when speed and strength matter more than perfect reconstruction. Stem cells renew specific tissues, bone rebuilds itself with remarkable patience, muscle adapts to strain, tendons and ligaments remodel slowly. The immune system defends, cleans and communicates. Cells repair, DNA, recycle proteins, and remove themselves when damage becomes too great. Even energy itself is part of repair. Mitochondria help produce ATP, giving cells the usable power needed to move, divide, signal, and rebuild. Nutrients provide raw materials. Sleep gives the body a quieter state in which hormones, immunity, metabolism, and brain maintenance can settle into a different rhythm. Across a lifetime, aging changes the speed and precision of these systems, while regenerative medicine tries to understand how repair might one day be supported more carefully. Repair is powerful, but it is not perfect. It can close a cut, rebuild a fracture, replace blood cells, calm inflammation, and preserve life through ordinary damage. but it is shaped by limits, trade-offs, age, environment, tissue type, blood supply, genetics, and the complicated history of the body itself. And perhaps that imperfection makes it more remarkable. The body is not a flawless machine. It is a living restoration project, always responding, adjusting, clearing, patching, and rebuilding. Even in rest, stillness, and sleep, it remains gently busy, maintaining the fragile conditions that allow warmth, movement, memory, thought, and life to continue. Quietly, constantly, beneath awareness, the body keeps doing the work of staying alive,